Yes, ma'am.
Good morning, madam.
Good morning, everyone. Good morning, sir. morning, Mundire madam. Good morning. Good morning. We'll just start in few minutes.
Madam, the music is too loud. We're not able
Yeah,
to
can
hear it.
you? Afrin?
Yes, we can hear now.
Yeah. Okay, Afrin, should we start now?
Yes, ma'am.
All right, let's start.
Camera on.
And keep the volume low. The music volume is too high. If you can reduce the
ma'am.
music
Yes, ma'am.
volume.
Yeah.
Okay, we can start. Yeah, over to you.
A very good morning and a heartfelt welcome to everyone gathered here today. I, Ms. Palak Vaira,
And I, Ms. Trisha Patil, together with our spirited and dedicated team of first year BPharm students, feel immensely proud to welcome you all to the 6th Student Research Congress 2025.
it fills us with great joy to see this meeting graced by brilliant minds, researchers, academicians, innovators, professionals, and aspiring young scientists, each of whom carries a spark that fuels the ever growing world of scientific discovery.
The 6th Student Research Congress is more than just an event. It is a celebration of curiosity, perseverance, and the shared belief that research has the power to transform society. Today, we gather not only to present findings, but to inspire one another, to exchange ideas, and to cultivate a culture of inquiry that thrives beyond these walls.
And now, as we move from the world of scientific exploration to the national spirit,
Let us rise in unity and honor the rich heritage and values that bind us together as Indians.
now we request everyone to please stand for the national anthem.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, everyone.
SVKM's Dr. Bhanuben Nanavati College of Pharmacy, an autonomous institute affiliated with the University of Mumbai, stands today as a symbol of excellence, quality education, and holistic student growth. With immense pride, we share that our institute is accredited by NAC with an A++ grade, and our BPharm program is NBA accredited till the academic year 2029.
Our college's commitment to academic brilliance is further reflected in its NIRF 2025 ranking, the 38th amongst the top pharmacy colleges in the nation and 4th in the state. With state-of-the-art infrastructure, a stimulating research ecosystem, and a faculty that nurtures innovation at every step, SVKM's BNCP continues to empower the next generation of pharmacy professionals and scientists. The institute has also earned prestigious government-funded research grants, strengthening its position as a hub for quality research with strong industry and academic partnerships.
Under the visionary leadership of our respected principal, Dr. Munira Momin, SPKM's BNCP offers a transformative learning environment, one that not only shapes careers but also builds character, creativity, and confidence of every student walking through its corridors.
The theme for this year's Student Research Congress is Innovations and AI in Healthcare. This powerful theme reflects the evolving landscape of modern science, where creativity emerges with technology and where the future of healthcare is being reimagined through data, design, and discovery. Over these 2 enriching days, we will witness remarkable contributions from young researchers through oral presentations across PhD, postgraduate, and undergraduate categories. Each presentation stands as a testament to the hard work, dedication, and scientific curiosity of our participants.
This year, we are excited to unveil 2 brand new categories designed to push the boundaries of applied scientific thinking. SRC Hackathon Oral Solids category and SRC Hackathon Artificial Intelligence category.
These categories mark a major step forward in blending hands-on problem solving with real-world pharmaceutical and technological challenges. We can't wait to see the innovative solutions and transformative ideas our students will bring to the stage. It is our privilege to extend our heartfelt gratitude to the University of Mumbai and Dr. Ravindra Kulkarni, Honorable Vice Chancellor, University of Mumbai, for the unwavering support and encouragement. Their continued guidance over the years has been instrumental in strengthening and uplifting this Congress.
We express our sincere thanks to our industry partners, Mr. Ajit Singh, Chairman, ACG Group of Companies, for graciously accepting our proposal to partner with this event. Dr. Ketan Parmar, General Manager, ACG Worldwide, and Dr. Parizad Elchidana, Principal Technical Consultant Pharma, ACG World, for consistently supporting the Student Research Congress year after year. And now, with great respect and enthusiasm, we invite our beloved Principal Ma'am, Dr. Munira Momin, to address the gathering and deliver the welcome address.
Thank you, Team BNCP, and very warm welcome to one and all today present on our SVKM's BNCP virtual platform. This virtual platform has become a reality since 2020. The COVID has taught us many things. One of the greatest things which we all learn is to communicate on virtual platform, which started feeling like it's in person. When we see each one of you here, let me request everyone if they can put the cameras on, and it makes us feel like we all are sitting in one room and talking to each other. My sincere request to all, if you can put cameras on, because that gives a more human touch, one-on-one. We feel like we all are talking to each other. So with that, thank you very much. Thank you, entire team BNCP, for pulling this off, wonderfully arranging and starting this. Most welcome, very warm welcome to Dr. Parizad Elchidana, our chief guest of today's event. Our keynote speaker, Dr. Santosh Dikshit. As my team mentioned, that this time we have started hackathon. Hackathon on AI, hackathon on innovation. We started this Student Research Congress in 2020 when we realized that there is a hunger for knowledge. There is a hunger for communication. There is a hunger to share ideas and spread knowledge. My student community used to constantly drop a message, even the parents. My student community used to constantly drop a message, even the my student community used to constantly drop a message, even the grow. Whenever I have met him and whenever I have requested him, he will simply say, "What do you need?" So thank you,
And whenever I have requested him,
ACG
he will
team
simply say,
for
"What do you
supporting
need?"
us. And it was University of Mumbai when we shared the idea with them, the Vice Chancellor then and the current Vice Chancellor, who immediately supported us for this event. And this sixth SRC, we have participation. I'm very happy to share that this time the participation is registrations are 850. Last SRC were 700 registration. In last SRC fifth, we had 361 institutes had taken part. This time in this year sixth SRC, we have 442 institutes have taken part. 235 universities have taken part, and I'm very happy. This is in the true sense national, because each state, all 28 states, has sent their abstracts and registration, plus 4 union territories. So when I look at the total states and union territory, there are 32 states and union territory total, and that shows that the Student Research Congress has gone across across India, and we are very happy. This time we have entries from US, Indonesia, and Nigeria. We have entries. Last year only one entry we had, and this time we have 3 entries from outside India. This talks about the spread of SRC. We have made sure that our experts are from across India, and we are grateful to all our judges who graciously accepted our invitation. Let me start with our chief guest, Dr. Parizad El-Chetana, who is a principal technical consultant of ACG Worldwide. She's the one who strongly believes, and I remember whenever I talked to her, she said, "Munira, tell us whatever you are doing new. I'm very happy to hear if any event or any opportunity where students are given platform to share their ideas." Thank you very much, madam, for accepting our invitation as a chief guest. She will also be one of the judge for our innovation. So this time hackathon is in innovation in healthcare and innovation in oral solids. ACG is pioneer. 93 to 95% of capsule business in the world is by ACG. ACG strongly believes in promoting oral solids, capsule being one of the biggest dose, one of the largest doses form contribution or a market which is captured by ACG. And we realize that ACG promotes innovation, and we requested ACG team that we would like to keep oral solids as one of the hackathon, and they were very happy. So thank you, Dr. Parizad, Dr. Parab, Rajesh Parab, and Dr. Subhashish, who three people would be representing ACG. Dr. Santosh Dixit. We always talk about innovation, and when we connected with him, he was very happy to graciously accept. So Dr. Santosh Dixit, who is a chief domain expert in the innovation lab, who else other than Dr. Santosh Dixit would be inspiring students, participants, upcoming generation with his concepts and with his ideas, and why the innovation From JSS, Dr. Anju Kamboj from Chandigarh. Just for all the participants, I must mention here that it is a student research congress, and hence, may it be UG, PG, or innovation or hackathon, we have made sure that it is research abstract which we will be accepting. So even for UG, we sometimes feel that there is no research at UG, but there are institutes, including ours. Research is as one of the important aspect we are promoting at undergrad level, where we have students working from second year, third year, and final year. And so a lot of institutes across India promoting research at UG level. So today we have wonderful abstracts, good quality research abstracts from UG, PG, as well as PhD students. In AI also, we have selected only the research, data-driven, outcome-based entries are selected, and hence our selection ratio is 10 point something, 10% selection ratio of abstract. So thank you very much once again, all those judges who have helped us in selection and who will be helping us today in selection. My sincere welcome to Dr. Parizad and Dr. Santosh Dikshit, along with all the judges of all categories. Thank you very much and have a wonderful 2-day session of knowledge sharing and spreading the innovation. Thank you very much. Over to you, team.
Thank you, ma'am, for the platform for this wonderful event with your inspiring words.
It is with immense pleasure and deep respect that we extend a very warm welcome to our distinguished chief guest, Dr. Parizad Elchidana, Principal Technical Consultant, Pharma at ACG World. Dr. Parizad Elchidana is an accomplished pharmaceutical scientist and a global industry leader with over 35 years of experience. She holds a PhD in pharmaceutical sciences from the State University of Ghent, Belgium, and is a certified ISO 9000 internal auditor with additional qualification in business management and computer science. Her extensive expertise spans R&D of patent, non-infringing products, quality management systems, and advanced product development using quality by design, QBT, and emerging AI-driven approaches. She has contributed to major global submissions including FTF, 505(b)(2), and NCE1, leading to several successful product launches in regulated markets. She also holds patents and has authored impactful research publications. Dr. Elchidana has held prestigious leadership roles, including Managing Director at Apotex Research Private Limited and Chief Technical Officer at Pharmax Pharmaceuticals, Dubai. Since returning to India, she has been serving as Principal Technical Consultant, Pharma at ACG, driving innovation and mentoring teams across the organization. Deeply committed to academia, she serves as visiting faculty, sits on multiple advisory boards, and is a PhD guide and examiner at several universities. She is also the president of the Controlled Release Society, Indian chapter, and co-chair of the AAPS Western India chapter. We are truly honored to have Dr. Elchidana with us today, and we look forward to the insights she brings to this forum.
Please join us in extending a heartfelt and enthusiastic welcome to Dr. Parizad Elchidana.
So thank you so much, and thanks for inviting me for this inaugural session of the 6th Student Research Congress. And I really appreciate the opportunity that Dr. Munira Momin and her team has given me to present today at this inauguration. Friends, it is a great pleasure to listen to Dr. Munira to the extent that the amount and the number of participants which has gone up over the period of years. This is really an exemplary achievement, and I really wish this complete research congress all the very best. So if I may, is it possible to share my slides?
Yes, yes. You can share, yeah.
Yeah. Can you see my
Yes. We
laptop?
can see
Yeah,
your
yeah.
slide
Yeah.
fully, yes.
Yeah. So it's an exciting topic, innovation and AI in healthcare. But what I would like to further extend is that AI in healthcare program will itself drive innovations in healthcare and make it a very fast track progress. Now, AI cannot be talked of as a standalone. It has to always be clubbed with machine learning. And this is something which I learned a very hard way. So AI and machine learning together have transformed the drug discovery. As we said earlier, accelerating the research and development timeline so that we are able to bring new and more affordable drugs at a very fast rate with the FDA approval. Not just that, during the current COVID situation, AI has helped with repurposing quite a few drugs. And this was the main reason why we could have newer drugs coming in for helping with the COVID related comorbidities, and they could be introduced at a very fast rate. Apart from AI in pharma, we are also talking of AI in healthcare today. And this is powered and transformed the medical diagnosis, personalizing treatment for patients, and also streamlining the various operations. And the most important healthcare innovations in 24-25 have been medical imaging, robotic surgery over here, where now they are using robots to carry out surgery. They call it microsurgery. And just giving you a case and example, where open heart surgery used to be a major surgery with the number of stitches has now gone into a microsurgery where just with a small hole in between the ribs, open heart surgery can be carried out. And imagine the recovery period of the patient. It is so fast and so quick with less amount of medication which is being given to the patient. So AI has really helped surgery and also sophisticated predictive analysis in patient care. Coming to remote patient monitoring and telehealth, this is once again the in thing today, which is come up with the help of not just AI, but earlier we used to call Internet of Things, which is now come to the extent of medical Internet of Things. And this again was possible only because of AI. And continuous monitoring of patients has now become possible through various wearable devices and sensors. And we can have monitoring of blood glucose levels, inhalations, which is affecting the patients, and sweat stickers. And this has not just helped the medical faculty, but has also helped pharma industry in a big way, that our clinical research has become so easy nowadays. We don't have to house patients. We don't have to house healthy volunteers as and when we are carrying out studies, clinical trials on our medication. We can just give them these wearable devices and monitor them remotely. Even operating efficiencies and automation has been a big help with the use of AI with respect to various administrative tasks that is being carried out, not just in various colleges, but even in medical institutions and hospitals. And predictive analytics for preventive care is once again a very, very big help which AI is given, because one can really forecast what is the potential about AI-powered chatbots and virtual assistants, which provide 24/7 discrete support for patients having mental health issues, and even counseling can be offered online. We spoke about accelerated drug discovery and development, wherein not just the identification of the various compounds which are simulating drug target interactions, but we can also predict toxicity profiles and even carrying out clinical trials and maybe bioavailability studies, bioequivalence at a fraction of a cost and at reduced time periods. So what is the industry outlook? Even keeping all these advantages in mind, I need to expand and state that although technology is advancing rapidly, challenges such as data privacy, algorithm bias, regulatory hurdles are key critical for us to move forward with AI-based systems, and we need to ensure that it's highly ethical and effective in its implementation. Now, coming to the regulatory agencies, how are they handling AI, and do they have any AI tools to support them? And the answer is yes. Just last month, it was reported that US FDA has now its own generative AI tool, which is called ELSA, and this is being utilized for fast tracking and reviewing the various submissions which are being made by companies. It also assists the FDA employees through various functions and even to investigate whatever data which has been provided and submitted by various pharma companies. Even to the extent that many a time submissions to the regulatory agencies are made in different languages, and it is now claimed by the regulatory agencies that the artificial intelligence supports them in translating those regulatory submissions in a very fast pace, and that's why decisions are coming in faster. So imagine, is AI the future or it's happening now? Friends, it's really happening now, and even today and tomorrow, we will be seeing a lot of AI-based initiatives that have been taken up in order to develop new products or in order to extend research. So AI is able to design new drugs. Not just that, it also gives us a big opportunity to design and develop new delivery systems, new formulations, new finished products. As I said earlier, clinical trials can be done at a faster pace. Drugs now need not be tested on actual animals for preclinicals. You must have heard of what we call currently as organ on a chip. So there are virtual models that are available which are there on a chip, and you can use those chips in order to check not just the efficacy, but even the toxicity effect of drugs, what effect it will have on the various cells which are present within the various organs. Robots not just help in surgery, as I mentioned earlier, they have started helping us in pharma manufacturing of medication and even their distribution. AI helps in process optimizations and controls. Batches can be released virtually. Testing can be done in the lab. The data can be picked up and the batch can be released. Counterfeiting of drugs is now becoming almost impossible through the AI-based systems like track and trace, which helps us in blockchain technology for the entire distribution channel. You can track even one single blister of the batch that is manufactured in the company. So this is to the extent which we have not just in pharma research, but even beyond research, where AI is really being helpful. So what is the future? In future, we can see the 3D printing of personalized drugs by local pharmacists. Why should someone who requires a 250 milligram dose take a product which is in 300 milligram? He or she can be delivered a 250 milligram dose by printing of that particular drug and giving it as a personalized medicine for that particular patient. We also see doctors have started printing organs, and one case study is there in the market where a heart has been printed and then it has been replaced. But unfortunately, it's done in an animal. It's a pig. The pig is still surviving. And hopefully, moving forward, we should be in a position to do it even for humans, and people requiring hearts may not have to wait for transplants. And coming to for the chips in a brain, which help not just the efficient learning, but will also possibly help patients who have been paralyzed to get back on track. And these are a few wearable devices which have been used not just for monitoring and managing patients or even healthy human volunteers during clinical trials, but as I said earlier, neuroformic edge computing on a chip and also systems application. Now coming to basic pharma, from completely using AI and ML from molecule to the market, these are the various stages which a drug goes through. And see, at each and every stage in today's day, we have something in AI which can help us fast track our product from molecule discovery to final supply chain in the market, where I mentioned about track and trace. Now, even on the shop floor level, manufacturing, we have smart machines. We have now what we call as continuous processing. And all these have been possible only through AI support. Under clinical trials, there are wearable devices, but one more thing.
Parizad, madam, you are muted by mistake, it seems. Unmute. Unmute yourself. Yeah, you're good.
So we have systems where we have end-to-end modeling and simulation, and over here, you need not actually carry out pharmacokinetic studies in human volunteers and patients. If you just know the dissolution profiling of your particular drug and the formulation, you can carry out in silico studies, and the software will tell you all. Coming to the regulatory and pharmacovigilance, which is very important in pharma industry. For the regulatory, I already explained to you, there is a software available with regulatory agencies. There is a software available with pharma companies who can collate the complete data which is required for submission to a particular regulatory agencies, and the software will make the complete submission and help you to submit in no time. So previously, which used to take months, now can be done in a few weeks. Pharmacovigilance reporting, yes, many companies, brand companies have this particular department. Currently, it is now a must, even in India, and pharmacovigilance also can be supported by AI-based softwares, where the issues that have been reported can just be put into the system, and AI will provide not just what the solution could be, but will also help in just sending the response back to the person who has reported the pharmacovigilance issues to the organization. Coming to product life cycle, I showed the complete product life cycle. This from end to end can be monitored and managed to excellent ERP systems and SAP. So SAP is a very, very old software which earlier used to just work on database, but now currently it is working on AI-based systems. So coming to AI in healthcare, what are the various myths and realities in developing medical for AI? People feel more the data, better the decision, and better would be the AI models. No, the answer is not the quantity of data that goes into it. It's the quality of data that matters. Only AI experts are needed. Sorry, just AI experts will not be able to do the work. People from various disciplines are required. It has to be a multidisciplinary team when you are building that particular AI model and when you are trying to execute and use it. And friends, this is the main reason why AI has now been introduced as a part of the syllabus during the B.Pharm course. So do take a lot of interest in this particular topic, in this particular subject, and it will take you places. The next point is AI performance leads to clinical confidence. Once again, you do get a little bit of confidence, but it requires a big validation that needs to be carried out. For example, we carry out various scans, various studies which are done on equipments. We carry out MRI, and what happens when the results come in? What is written at the end of the complete report? Have you ever seen that? The results are declared through AI and through the software, but basically at the bottom, it is always written that it is to be correlated with clinical findings. And there you go. You can't just depend on that data. You have to go back to the clinicians, and he will be the best judge to give you the final interpretation of those particular reports. Integrating AI into routine workflows is straightforward. No, friends, it's not straightforward. AI may be artificial intelligence, but it's always the human mind. It's always the human brain, which is going to finally look at all the data and take decisions moving forward. Launch of AI means success. Yes, launch of AI would mean success. It will give you high quality performance through continuous monitoring and iterations. But once again, human brain is what is required behind the artificial intelligence. So to summarize, it helps fast tracking development. It provides remote access. It promotes multicentric studies, supports predictive analysis, enhances decision-making capabilities, and complements continuous improvement, but it has to be hand in glove with the human mind. Implementation is a challenge. People with the right skill are required. You need the necessary infrastructure. As I said, AI standalone is not effective. Machines are required along with the AI softwares in order to make them work. Finance is very key critical. All these things are high cost, and regulatory compliance is very important, because just using an AI software will not be acceptable to the regulatory agencies. It has to be properly validated and proven that it is doing what it is supposed to do. So let's start slow. We have started, we have picked up the pace, and it's now time to move forward. So thank you very much. Let's keep the India flag flying high. We are the pharmacy of the world. Let's be the innovative pharmacy of the world. So thank you very much. And over to you, team.
Thank you so much, ma'am, for that inspiring and thought-provoking address. Your words have truly resonated with each one of us, and we are deeply grateful for the valuable insights you've shared today.
Now, it is my privilege to invite our next keynote speaker,
Thank you so much. Thank you.
Now, it is my privilege to invite our next keynote speaker, an eminent expert who stands at the forefront of innovation in healthcare and life sciences. We warmly welcome Dr. Santosh Dikshit, Chief Domain Expert, Healthcare and Life Sciences, Innovation Labs, Persistent Systems. Dr. Santosh Dikshit, an accomplished scientist and thought leader with over 18 years of diverse experience spanning drug development, translational oncology, pharmacogenomics, and healthcare innovation. Dr. Dikshit currently serves as the Chief Domain Expert, Healthcare and Life Sciences at Persistent Systems, where he leads business development, thought leadership initiatives, and capacity building efforts, driving impactful solutions at the intersection of healthcare and technology. Prior to this role, he worked as a consultant, contributing his expertise towards strategic planning and innovation in the life sciences domain. Before joining Persistent Systems, Dr. Dikshit played a pivotal role in establishing and managing the Center for Translational Cancer Research, Pune, where he developed a comprehensive breast cancer-focused research program. His earlier work at Prashanti Cancer Care Mission involved designing public health initiatives for community-based breast cancer awareness, setting up genetic counseling units, and developing clinically relevant research projects. across science, regulatory strategy, clinical research execution, and healthcare innovation. His analytical acumen, strategic vision, and multidisciplinary expertise make him a transformative leader in healthcare and life sciences space. We are privileged to have him with us today, and we look forward to the insights he will share from his remarkable journey across academia, clinical research, and healthcare technology.
Can you please confirm if you see my slides and if the audio is okay?
Yes, we
Yes.
can see your slides, sir, very clearly.
Okay. Good morning, everybody. And at the outset, I'm deeply grateful to the Nanavati College of Pharmacy and the principal madam for this opportunity. Deep at heart, I'm still a pharmacy student, despite being in this career for a long time. Every opportunity to meet students, either online or in person, is something that we cherish. And more so with this topic, which is probably very close to heart. This is also bread and butter for me today. There are tremendous disruptions happening. Perizad madam has laid the foundation very well. And she has covered a lot of topics, which are really the ecosystem topics of today. I'm going to go a little deeper into the same topics, and I'm trying to give some examples of how this industry is shaping up and how our conventional pharmacy education very soon is getting outdated. And my key take home at the very beginning is that I think we really have to adapt with the times right now. The old ways of doing things, the old way of learning, I think those are getting completely outdated. And unless we keep pace with all of this, the AI disruptions, we are probably going to become a little bit outdated and obsolete, etc. And we don't want to do that. So what I want to convey to you is, being in the industry and practitioner of AI, I'm going to mainly focus on a couple of real life use cases and our own observations of the industry, right? So for the next 20 minutes that have been allocated, I'll be taking a more industry focused approach here. So let us look at how the industry is shaping up. And I'm using this.
Dr. Santosh, your slides are not visible now. Dr. Lokesh, do you want to connect with him? Because I think he got disconnected also, it looks like.
Yes, ma'am, I want that.
Just bear with me. I'm back.
Okay. Yeah, we can see your screen.
Yeah, okay.
Yeah.
So
we can
on your
see your
left-hand side is the emerging technology. I think as practitioners of this field, we don't really have to go into technology details. We have engineers, computer scientists, all the technologies doing all the wonderful work, but we need to understand the vocabulary. So today we are going to mainly focus on artificial intelligence, big data, but soon there is quantum coming up. There are new technologies that are constantly getting into this first pillar. I think the second pillar is where all pharmacists, all of us are very deeply involved with. One is for us to understand the changes that are happening in core medicine. There is a very popular term that is used today.
It seems it's once again disconnected. Just see, Lokesh.
Yes, ma'am. Let us check.
I think she has. I think he has some connection.
Is the problem at your end or is it at my end?
No, I think could be because here everyone is there.
Okay.
I think your computer is getting disconnected, something like that.
Okay. If that happens, I'm going to switch my network. Just bear with me. But let's
Sure.
continue on this right now.
Yeah, we can see now.
Yeah. So now the green pillar is where there are tremendous changes happening in medicine. The medicine is becoming P4, that is preventive, participatory, predictive, personalization. There is tremendous amount of genomic revolution that is coming in. And all these changes are reflecting in the pharma industry, where we are seeing this disruption. There is a new term which is called as Pharma 5.0. This is a term that is becoming very popular, where you are seeing the interface of human and AI coming together into a lot of these activities. There is a background. Can somebody mute?
Yes, we have muted them. Dr. Santosh, Satish, please, Dr. Santosh, go ahead.
You can hear me, right?
Yes, we can hear you.
And you can see my
We can't
slides?
see your. Yes, yeah, we can see your slide
Okay.
too.
And the third pillar is what is happening all over the world. Of course, we have to learn from it, but we have to adopt it to local requirements, right? For example, a genomic biomarker in Caucasian population may not be useful for an Indian population because of the pharmacogenetic differences, etc. So that is where one example of global to local calibration has to be done. Second is when you think of technology readiness, a technology readiness in Europe or America could be completely different than in India. In fact, India has a major advantage in our digital penetration with 5G and 6G coming up. So we might actually be doing better than the West. You have seen how we are doing very well in digital payments and etc. Right? So that's another advantage. The third advantage that we have to think about is the regulatory environment that is happening in the US versus regulatory in the Indian ecosystem, etc. So you need to constantly think of the global trends but adapt to the local trends. And the last part is where you think of business strategy. How am I going to justify investment in technology, return on investment? Who should be my partner? Is this going to be a short-term or a long-term investment, etc.? So these are typically the thought processes that go into adoption of AI. Now, if you look at our biopharma industry, I think this is a landscape view of the industry. At the center is the funnel, the drug discovery funnel, where we start with medicinal chemistry, creating hits and leads, and then going through the entire process of drug discovery and development, right? So this funnel is going to be fundamentally intact. But what is changing is the allied industry to a funnel. So if you look at scientific instruments, medtech devices, diagnostics, lot of contract research organizations, etc. This industry is proliferating because of the data explosion as well as the AI adoption. And on the right hand side, the technology industry is also exploding because of availability of what is called as an AI toolkit. And this AI toolkit is where you are seeing large amount of technology companies getting into the pharma space. In fact, there is a new term that is being used in the industry. This term is called as tech bioservices. Please note, this is not biotech, but this is tech bio. So for example, a company like Persistent, despite being a data and tech company, they are investing in opportunities in the biopharma space. So that qualifies for us to become a tech bio company. And what a tech bio company does is it brings product engineering, it brings data infrastructure, and it brings a lot of this AI toolkit to the ecosystem that we are all familiar with, right? So this is a major trend that you are seeing right now. And then the biggest question is, what are the principles of AI adoption? How do you think about AI adoption, right? So all across the industry, principally, there are 5 major thought processes. The first thought process is it's a top-down approach. That means there has to be a robust leadership-driven AI R&D strategy. This cannot be just a one-day thing. This has to have a long-term vision coming from the CEO, the board of directors, and then trickling it down into the organization. And that also involves creating monetary infrastructure as well as technology infrastructure. That is where your second layer comes into picture. So a lot of pharma companies are investing into futuristic data foundations. These are to modernize your existing data processes, to create what are called as cloud services or on-prem services, to protect your data, to clean your data, etc. So all this becomes what is called as a data foundation. The third is judicious choice of AI models. And I think Perizad madam mentioned that it is not always AI ML. It could be generative AI. It could be agentic AI. It could be future generative intelligence. A lot of these different AI versions are coming into picture. When to use which AI is a very, very important choice because it determines the output. It determines the cost and speed of operations. Number 4 is now you have selected your data. You have selected the model. How do you implement these models into workflows with best practice adoption? So interestingly, US FDA has come up with a guidance which is called as good machine learning practices. we have all heard of good clinical practice, good manufacturing practice, good laboratory practice, but now there is good machine learning practice. That means whatever you do has to be done with regulatory standards. And the last and the most important point, in my opinion, is constant learning. And even at this stage in our careers, we have to constantly learn, not only learn, but unlearn and relearn. So that is a new process. And I'm very happy about the theme of this conference, which is AI innovations, commitment to learning, etc. Right? Learning doesn't stop. We are all lifelong learners. And that is exactly the attitude that the industry is seeing right now. Now, if you see broadly, if you break down a pharma industry, there are different units of pharma industry. Let's say preclinical research, clinical, commercial launch of products, regulatory compliances, medical affairs, then enterprise technology services, and overall corporate functions. There is not even one unit which is untouched by AI today. I have just taken a couple of examples of how these industries. I'm going to go a little bit deeper into the R&D part. The biggest disruption that is happening, and already Perizat madam mentioned that, is in the field of research. The conventional model of 10 years of research, more than $1 billion investment, I think that model is getting completely disrupted now. You are seeing tremendous expedition of the journey of drug discovery and at low cost, etc. Right? So for today, I'm going to mainly focus on R&D and not other functions. So when you look at R&D in today's world, whether you are a multinational corporation, whether you are an Indian pharma, whether you are a large enterprise, whether you are a startup, these are the 5 or 6 major areas where you see tremendous disruption coming through AI applications in R&D. The first is explosion of genomic data. We all call it multi-omics, genomics, transcriptomics, proteomics, all kinds of omics. The second big growth engine is in the area of biomedical imaging. In fact, if you look at the largest number of FDA approved AI models, they happen to be in the area of biomedical imaging as of today. And biomedical imaging could be imaging of the cell, imaging of the tissue, that is called histopathology, imaging of the organ. It could be preclinical imaging of the animals, etc. So that is the complete gamut. The third is what we call as preclinical R&D, mainly computational biology, computational chemistry, where we do drug repurposing, de novo drug design, in silico, medicinal chemistry, etc. And number 4 is where already we heard a little bit about using AI models to obviate the need for animals. This is where there are virtual cells coming up, there are virtual animals, virtual organs on a chip coming up. So these are all enabled by AI. On your left-hand side is human clinical research. For example, can I design a better trial with the help of AI so that I have the right choice of the patient for the eligibility criteria in a trial? This is a very important problem that AI is solving in the CRO industry. Number 6 is pharmacovigilance. For example, when your product is in the market, whether it is a diagnostic product or a drug, you generate a lot of data, and that data is called as real world data. Can we use that data to improvise the performance of the product in its second and third generation? And number 7 is personalized medicine, where you use either a imaging biomarker or a genomic biomarker or a protein biomarker, which is from your own biospecimen, in order to determine what is the current state of the disease, what may happen in future, could there be remission of the disease, etc. These are all significant areas where the pharma R&D industry is getting impacted by the AI onslaught, right? Before I go to use cases, I just want to tell you how the industry develops AI digital platforms. I would like you to look at this diagram bottom up. At the bottom is data. I emphasize the importance of data. And in order to understand data, you also need to understand fundamental science, fundamental biology, clinical medicine. So the dominance of domain understanding is paramount. It's a foundation layer. The second is called data engineering, where you bring in data from various sources, and then you pass this data through what is called as data curation engines, data compliance engines, data governance engines, etc. And here you need to understand the data lake, or let's say the data manufacturing concepts as such. The third is the technology stack. This is where companies like Persistent or Tata Consultancy or any other company, they actually excel. They provide a lot of these data services, technology services, so that you maintain this data at scale over a perpetual period of time with data security protections, etc. Having built these technology, data, and domain blocks, Now you start developing applications. I have listed few R&D applications which are impactful. There are significant startups being generated in this space. Many startups are only focusing on organoids, etc., and creating very interesting, unique innovations. And then once you start developing these R&D models, you start seeing impact at a translational level, that is at a strategic outcome level. So sometimes you start developing new biomarkers, completely new cohorts for your drugs that you are developing, and that results into business impact. So you can probably think of new business models to monetize these models in order to increase your R&D productivity. You can think of drug repurposing as a business impact model, etc. And together, this industry therefore serves the pharma ecosystem, as we call it, or the healthcare ecosystem, right? So please remember, data is very important. Understanding of data engineering. You don't need to understand technology, but you need to understand the vocabulary, and then you need to be good at what you're doing as a pharmacist, scientist, etc. Right? I'm just going to take a couple of examples. The biggest disruption that you see already happening is in the field of computational drug discovery. If you see the entire journey of drug discovery, it starts with mining large amount of biological data for identifying de novo targets. Then, because everything is genome related today, you see a lot of mutations. Then, if you identify your gene of interest and the protein of interest, you need to do structure predictions. And with the structural biology construct of your gene of interest, you start developing gene molecules, chemical molecules, etc. And then you go to the hit to lead ratios and property predictions for ADME, etc. So this complete journey is now getting expedited because of advent of AI. At every stage, you have very powerful AI models. And if you follow this field, please look at companies like In Silico Medicine or Recursion Pharmaceuticals, etc., who are trying to disrupt this space by short-circuiting a lot of these rules. And here you see companies like Google, Microsoft, Nvidia, they are investing significantly to power this kind of a journey by their AI models and infrastructure coming together. So this is going to be a very productive area for all of you if you are interested in computational drug discovery. You will see tremendous opportunities not only for career, for innovation in this particular area as we go forward. So my own group, we work in this area, and we have done something called as developing a knowledge graph for Crohn's disease. Crohn's disease is a inflammatory bowel disease, which is a precursor to colorectal cancer. What we have done is, with the help of AI, we have created a knowledge graph for one of our pharma clients. In that knowledge graph, we take a lot of clinical data available through the client data sets and the molecular data. We clean that data with the help of Gen AI. These are called as large language models. Everybody knows LLM today, ChatGPT, Gemini. But the real output is for a scientist, where we have this interactive knowledge graph in the form of a relationship between all these data sets. You can see this family tree relationship. And more importantly, I empower this knowledge graph with the help of a chatbot. And that chatbot can talk to this knowledge graph. It's a very complex knowledge graph, but the chatbot can give you authenticated information with primary literature, with a lot of data access, with confidence scores, with explainability of the AI. So whatever output I get from the chatbot is related to the data that I'm looking at, but it also tells me the logic of how that response has come out. So this is very popular in the industry right now. This is called as a AI-powered knowledge graph. And I think you should really, as students, you should track this field. Knowledge graph is going to be a very popular theme even today and even in future. The second area I want to briefly touch upon is the area of clinical trials. What I have done is I have broken down the entire life cycle of a clinical trial into 3 phases. The first is called as a startup phase. That is before you actually dose a candidate on a clinical trial. There are a lot of activities like clinical trial design, statistical analysis plan, writing a protocol, getting the approval from the ethics committees, setting up the budget, contracting, etc. The second part is the actual implementation, where you choose the right patient, you monitor the patients, you collect the data, etc., and then you close the trial. And in the third part is where you have to submit the trial data to regulatory agencies, you have to write reports, you have to write manuscripts, and then you have to close out the trial, etc., right? And then there is a constant need for training together. Almost each one of these activities is now getting impacted positively because of AI. So you can design a better trial, you can do a better statistical design plan, you can actually write better documents because of AI. This is a big cottage industry today, AI guided medical writing. There is a tremendous need for domain scientists to understand how to validate the medical writing generated by AI. And these are going to be, again, big career opportunities in the CRO industry for each one of you. Taking one example, me and my group, we have worked with a large pharma company with global presence, and the use case was very interesting. They only had 70 medical writers, but they were not able to meet the demands of their global medical writing department. So they came to us saying that, can you help us design what is called as a draft zero concept? Draft zero is essentially a generative AI generated medical writing draft that can be further validated by human medical writers. But the advantage here is writing a draft, if it requires 5 working days, I could potentially do it in about 5 minutes. And that is the beauty of Gen AI guided medical writing. And we were able to successfully deliver this solution for the pharma medical writing unit. And they gave us feedback that they are seeing 80% increase in the speed of writing this, about 40% improvement in the accuracy, and then time saved is money saved. So there was an economical advantage of developing this kind of a protocol. And we created the first use case for clinical trial protocol drafting. But you can imagine every document that can be submitted for US FDA regulatory can undergo this kind of a treatment. So this is a very popular use case right now. I want to switch and go to the actual area of patient care. We call it precision medicine in today's world. Remember, I told you about P4 medicine. So what happens in precision medicine? It is no longer one size. Okay. So precision medicine is not one size fits all, but it is individualizing therapy according to your biomarkers. So a futuristic hospital is going to look like something like this, where you have input consultation screening. There is conversational AI that will enable a physician-patient interaction either remotely or in person. The output will be converted into electronic healthcare records. So you don't have to go back to old methods of taking notes. The advantage here is the physician can give quality time to the patient and not worry about taking notes. And then furthermore, if you
Dr. Santosh, you're muted. Dr. Santosh, you're muted.
Yeah,
we can hear you.
Okay. So now a futuristic hospital will look something like this, where every process is going to be AI derived, and there will be a collaboration between human experts and agents to deliver basic functions. Those basic functions will be orchestrated by what is called as a master control agent, almost like a head of the department, or let's say in this case, Munira madam. And then you will have reporting agents that will synthesize all this information and then pass it on to the healthcare professional, or pass it on to the patient, or even to regulatory agencies, etc. And this is no longer fictional. This is actually happening as we speak. In United States, we are working with a lot of hospitals who are trying to utilize our services to create these kind of workflows. And even in India, we are talking to some large hospitals who are trying to adopt what is called as agentic AI workflows according to the regulatory standards, etc. So anybody in this group who is doing PharmD, a clinical pharmacy residency, working with hospitals, etc., you are going to see a lot of AI adoption according to what is called as agentic AI workflows, etc. So this is a very futuristic theme, but this is going to hit us very quickly. And I think Perizat madam talked about a very important aspect of regulatory impact. So what is happening today is the conventional model of regulatory oversight, where large amount of information in a printout, thousands and thousands of pages is sent to FDA, and then FDA takes its own sweet time to review, and it comes back, and then it again takes a lot of time to do iterative reviews, etc. So this model is getting completely disrupted now. Generation of documents, review process, sponsor guided AI solutions, FDA guided AI solutions. This is how the field is moving right now. So there is AI tools available to the sponsor. There are AI tools available at the FDA level to review the processes very well. And you will see tremendous amount of what is called as efficiency gain in this entire process of FDA regulatory reviews coming together. And rightfully, Perizat madam mentioned about this tool. I would very strongly encourage the students to follow this tool. This is going to be a major disruption in the field. The tool is called ELSA. It is an acronym for Electronic Services language assistant. What this tool does is basically it summons all the information that the FDA has received. And with the help of a chatbot, it will help you understand certain questions that you can ask in the form of a prompt. So you can imagine like a chatbot or a chat GPT at FDA, but here with scientific data. And currently what is happening is these are the use cases of FDA. FDA can do clinical protocol review. It can do scientific evaluations. It can do pharmacovigilance summarization. It can help you compare labels between two different drugs. It can also help you identify the site selection, site inspection, manufacturing site reviews, etc. And mainly reading, writing, summarization. So this is actually happening as we speak. This is no longer theoretical. And there are guidances coming up about how ELSA needs to be adopted by the sponsor so that there is streamlining between the sponsor submissions and the ELSA receipt, etc. together. So anybody who wants to make a career in regulatory, please remember you are going to work with a lot of these GenAI tools as we go forward together. I want to conclude with 2-3 cautionary notes. There is a hype in AI. Just like any industry, people are trying to make the most of AI. So there is a hyper bubble. At some point of time, that bubble is going to stabilize. Regardless of that bubble, AI adoption is real. There are tremendous impact areas that are coming up. I just showed you a few examples. But any AI without a responsible AI framework is actually a double-edged sword. In fact, it will harm you rather than benefit you. So one very important aspect for anybody who wants to get into field is to understand the responsible AI elements, which are defined by ethical, legal, societal, and intellectual property implications, right? And for that, you need to understand data governance. You need to understand data protection, data privacy. Government of India has recently released what is called as a Personal Data Protection Act. We are all aligned with the Government of India AI regulation initiatives, and these are aligned with the global initiatives. There are best practices of data privacy, data protection. We need to understand them. Like I mentioned, there are regulatory guidances coming up from the industry, which are called as CGMP guidelines, GXP guidelines. This will come into the regulatory parlance. And more importantly, all this data has to be protected because you may be dealing with patient data. So cybersecurity and all those things become very, very meaningful together. So understanding responsible AI elements for a pharmaceutical scientist is no longer an option, but it is a mandatory thing. And this also can become a career track in future for anybody interested in what is called as responsible AI, digital trust kind of career paths together. So I'm going to conclude. This is a bit of my own call for actions because I see a lot of students here. It is important for you to follow these tenets. It is important for you to become multidisciplinary. Unidimensional knowledge in today's world is going to be outdated. But where do you start? You have to start with small use cases. And I think the Innovation Hackathon today is a great start. But you also have to constantly... Sorry?
I didn't give me my phone back.
Are you saying anything to me?
No, I think there is somebody's background noise.
Okay, all right. So first, identify use cases, talk to the industry experts, talk to real practitioners, interact with doctors, interact with pharma scientists, interact with AI scientists. Second is invest in understanding a lot of data infrastructure. And I'm very glad that there is a curriculum coming up on AI, but that curriculum should also sub-verticalize in data practices. So madam, that is a suggestion to you that also talk a little bit about biomedical data, etc. Very important. I just mentioned the importance of ethical and cultural alignment with technology and AI. This, in fact, if not done well, this can work against you. And then you have to be pragmatic. You have to start somewhere. Maybe think like a smart startup. Do one thing, but do very well so that it can scale up in future. And then constantly invest in self-learning, self-education, organized learning, institutional learning, etc. Because this is a dynamic field. The AI concepts are getting outdated with literally within months because it's a hyper competitive area right now. So an AI model which was very popular a month ago may not be popular. Somebody else must have trumped that model with a better model, etc. Right? So this is a very, very dynamic field, etc. But at the same time, this is a very exciting field. The pace of innovation, the pace of adoption, the pace of regulatory compliance, etc. It is phenomenal, and we all cannot be untouched by what is happening here. I would like to thank the organizers for the opportunity. I can interact now if there is time, or you can always reach out to me. I'm very happy to continue this conversation further. Thank you for the time here.
Wonderful, Dr. Santosh. Your talk and Dr. Parizad's talk, they were so much very well aligned and touching the core of the entire philosophy and the entire world where it is going. I just wanted to share with you that our president of SVKM, he mentioned this time that each class must have the AI concept. And that's where we started at our college from this year. AI basics, AI in pharmacology, AI in chemistry, AI in formulation development, manufacturing, where we have started from first year B Pharm to final year B Pharm, as well as first year M Pharm and final year M Pharm. So you rightly mentioned that, yes, it's a time we need to sensitize students towards AI, and we need to not train, but at least give them concepts so that they can make career in future, because AI is the future. Thank you very much, Dr. Santosh and Dr. Parizad. Very aptly shared your thoughts and preparing, or I would say inspiring, a student community and the research community towards future. Over to you, team.
Thank you so much, Munira ma'am. Thank you so much, Dr. Dixit, for your insightful and deeply enriching address. Your perspectives on innovation and the future of healthcare have truly broadened our understanding and inspired us to think beyond conventional boundaries. We are grateful for the clarity, vision, and motivation you have shared with all of us today. We also extend a warm welcome to our highly esteemed panel of judges whose experience, insight, and commitment to nurturing young researchers elevate the spirit of this conference. Your presence today reflects your steadfast belief in fostering research, innovation, and scientific growth among students and scholars. We are deeply grateful for the knowledge, inspiration, and direction you bring to this event.
Thank you, everyone. We now request all participants to proceed to the designated presentation room.
Just a second. There is a student, there is a lot of echo. There are 2 devices logged in near you. May I request chief guest and the keynote speaker and all the judges who are present, if they can put their camera on, we can have a screenshot of that. May I request all the judges as well as the participants, those who have joined, if you can put camera on, and students, if you can move this flyer, the poster which we have put up right now, so we can have an entire grid of all the
Yes, ma'am, just a second.
yeah, we can take a group picture. We can request all the members if they can put the camera on. We can take a group picture. See, every event at the institute, even at the industry, we always go for finally a group picture, because the group picture becomes the memory. So may I request all the members, judges especially, judges, participants. All right, somebody can probably take a screenshot.
Yes, ma'am.
All right. Perfect. All right. Thank you. Thank you very much, Dr. Santosh and Dr. Parizad. Thank you very much. It was a wonderful listening to both of you.
Madam, I'm going to drop off because of work commitments, but please reach out to me. Very happy to interact further. And we do come to Andheri. In fact, we have an office in Marol. So if there is opportunity to meet any one of you in person, very happy to do that, Ines.
Sir, wonderful. We will connect with you after this event. We'll drop you an email and we'll see how at a common convenient time, we definitely would like to invite you as per your availability. We'll be more than happy. My team would be very much happy to interact with you.
Yes, likewise. Looking forward. Thank you, madam.
Yeah.
Thank you, everyone.
Thank you.
Thank you.
Thank you, Dr. Parizad, and thank you all the speakers as well as judges. Thank you very much. Over to you, team.
Thank you, everyone. We now request all participants to proceed to their designated presentation rooms and kindly ensure that you enter using the allotted codes as your presentation leads. Please note that displaying personal names or institutional affiliations in any form will lead to disqualification as per the event guidelines. To all other attendees, we invite you to join your respective rooms as well. For those who wish to view the sessions online, the event is being streamed live on our college YouTube channel, SVKM's BNCP. You may also visit our official SRC website, bncpsrc.com, for additional details and ongoing updates. Respected judges, we request you to join the rooms assigned to you as mentioned in the official email. Should you face any technical difficulties, please inform us immediately so we may provide prompt assistance. Thank you once again for your cooperation, participation, and support. We look forward to an engaging and enriching set of presentations ahead.
Can we, I request Afrin Madam, if we can put both the team links in chat. So if some of the participants who do not have the link for those rooms, they can join directly
yes,
from this
yes,
link
ma'am.
itself.
Yes, ma'am. I'll share it on the chat box here as well as it is shared on the WhatsApp group and email as well. So I'll share it here on the chat box.
Yeah. So if those who have not received email
Yes,
and joined inaugural, they can join respective.
yes, yes, ma'am. Yes, ma'am. I'll do it.
All right. Perfect. Thank you, everyone. Let's meet into UGPG rooms. Parizat Madam will be more than happy if you wish to join any of this room. Do let us know. The link is being shared into chat box. Soon we will share, and then we would be more than happy to see all the senior members and everyone in either of the group.
Yes. Dr. Munira, is it possible to get the list of the topics maybe which are being presented today so that accordingly we can join the rooms?
Okay, Afrin Madam, if you can do that.
Yes, ma'am.
Yes.
Yes, ma'am. I'll share it.
Right. Okay,
Okay,
thank you.
share the Parizat Madam, but share the meeting link over here, please.
Okay, ma'am.
Meeting link,
So-
I think we have in the agenda which was shared yesterday.
Yes.
Yes,
Yeah,
ma'am.
the links are
it's
there.
there.
Yeah.
We can share here also if anyone who has not received the email. Because this link was open, there are some other members who have not registered may have joined. And if they
Right.
wish to listen. Yeah.
Right. Okay. Thank you so much. We'll be there.
Okay. Thank you very
Yeah,
much.
thank you. Thank you.
Thank you.
Thank you. Thank you, ma'am.
Thank you.
Actually, list is there on the website also.
Yeah, the link is on the website. Afrin, okay, room 1 UG, share so that I will join from here itself. And keep this room on in case if anybody wants to join at the later stage, they will have these links.
Ma'am, the same room we continue for undergraduate presentations. Only postgraduate presentations will move to another room. So the
Okay.
postgraduate link is there in the chat box. All undergraduate presenters will stay in the same room, same meeting.
Okay, yeah, just give the clarity so people know it.
Yes, yes, ma'am. Yes, ma'am.
I will just welcome the judges of UG and then switch to PG.
Yes, ma'am.
Dr. Daivat, Dr. Kavita.
Good morning, ma'am.
Good morning, Dr. Kavita. How are you?
Fine, ma'am. Doing good. How are you, ma'am?
I'm good. Thank you very much for joining us.
Thank you so much for the opportunity.
Thank you. Dr. Daivat? Afrin, can you put the Student Research Congress flyer instead of participants?
Yes, ma'am. Madam, Kavita madam, we'll be sharing the 6th SRC flyer with you. This is past years. I'll share it with you shortly. You can use that for the frame.
Okay.
It's okay.
Afrin, do let me know if PG judges are already there, then I can join that and come back here.
Okay, ma'am, we'll just check.
Dr. Daivati is here?
by SVKM's Dr. Bhanuben Nanavati College of Pharmacy, Autonomous, in collaboration with University of Mumbai and supported by a valued industry partner, ACG Worldwide and SciTech Center.
Science knows no country because knowledge belongs to humanity and is the torch that illuminates the world, a profound thought expressed by Louis Pasteur and the one that truly captures the essence of what brings us together today. This sentiment beautifully reflects the spirit of a gathering where young minds come forward to share their ideas, spark innovation, and contribute to the ever-growing body of scientific knowledge.
This session celebrates the curiosity, creativity, and innovative thinking that undergraduate students bring to research. This introduction of this category aims to ignite a research mindset earlier in the students' academic journey, encouraging them to question, explore, contribute meaningfully to scientific inquiry. Through this initiative, we hope to nurture a foundation for critical thinking, deeper learning, and future research pursuits, inspiring students to see research as higher education and exciting avenues filled with opportunities.
We are also honored to welcome our esteemed panel of judges whose expertise, guidance, and thoughtful evaluation will enrich today's session and motivate our young researchers. The judges for the undergraduate category are Dr. Kavita A.N., Dr. David Parekh, and Dr. Rashmi Malia. We would now like to introduce them. Judges, can you please switch on your cameras?
Dr. Kavita A.N., an accomplished academician and researcher, is currently serving as the Vice Principal, Professor, and Head of Department of Quality Assurance at Krupanidhi College of Pharmacy, Bengaluru, with an impressive 18 years of experience in teaching and research. Dr. Kavita has established herself as a leading expert in nanotechnology-based drug delivery system and pharmaceutical product development, especially through the Quality by Design, QBD. She has significantly contributed to academic growth by guiding 24 M.Pharm research students. Her research encompasses self-emulsifying system, lipid-based formulation, in situ ophthalmic gel, fast-dissolving buccal delivery system, and analytical method development using QBD. She has authored over 20 work has been widely recognized at both national and international platforms through multiple oral and poster works. Dr. Kavita also plays a pivotal role as managing editor for Journal of Pharmaceutical Research and serves as the IQAC coordinator, demonstrating her commitment to academic excellence and institutional quality. She is frequently invited as the resource person for workshops, national webinars, and faculty development programs, especially in the area of QBD and pharmaceutical quality assurance. Her dedication to innovation, quality-driven research, and professional mentorship continue to inspire students and researchers alike. We sincerely thank Kavita ma'am for taking the time to join us today and for gracing this event with her esteemed presence.
Thank you so much. I would like to thank BNCP team and Munira ma'am for giving me this great opportunity. Thank you so much, ma'am.
Thank you, Kavita madam. Thank you for accepting our invitation. Yeah, over to you, team.
Thank you, ma'am. Today, we also have with us Dr. David Parekh. Dr. David Parekh is an accomplished assistant professor in the Department of Pharmaceutics at the Institute of Pharmacy, Nirma University, where he has been serving since 2006. He completed his B.Pharm from KBIPR, M.Pharm from LMCP, and PhD from Nirma University. He has been honored with multiple institutional awards, including the Best Assistant Professor Award 2015 for innovative teaching and recognition for his outstanding contribution to alumni engagement and university web development. Dr. Parekh's research focuses on novel drug delivery systems, particularly gastroretentive systems, oral cavity targeted formulations, medicated chewing gums, and topical nanoformulation. He has published over 10 research papers in reputed journals such as the Journal of Controlled Release and Expert Opinion on Drug Delivery, and has presented his work internationally in Malaysia and the UK. He is a life member of several professional bodies and currently serves as the Vice President and State Level Career Guidance Counselor of IPGA Gujarat State Branch. He is also sought after speaker, having delivered numerous invited talks in STTPs, FDPs, conferences, and career development programs. We are delighted to have Dr. Parekh with us today and deeply appreciate his presence. Thank you for your gracious presence and for dedicating your time to this event.
I like it. So cute. तेरे पास भी है ना?
I would also like to introduce Dr. Rashmi Malia. It is our pleasure to introduce Dr. Rashmi Malia, Associate Professor and Head of Department of Pharmacognosy at SVKM's Dr. Bhanuben Nanavati College of Pharmacy, Autonomous. With an enriching academic career spanning 21 years, Dr. Malia has made remarkable contribution to the field of pharmacognosy, particularly in herbal formulation development, phytochemical analysis, and the biological evaluation of medicinal plants. She has guided over 35 MPharm students, nurturing young researchers and advancing scientific inquiry. Dr. Malia has made an impressive publication record with 56 research papers in reputed national and international journals, along with the authorship of 5 book chapters, reflecting her depth of expertise and commitment to the scholarship. Her achievements include the prestigious Dr. P.D. Sethi Memorial National Award 2016 for Best Research Paper. Under her mentorship, students have excelled at the university level competitions such as the Avishkar Research Convention, and the department has earned recognition, including a 3rd rank in BRPR 2025-26. Dr. Malia has also secured government and industry funded research grants amounting to Rs 5 lakhs, supporting innovative work in natural product research and herbal development. We're truly honored to have Rashmi ma'am with us today. We extend our heartfelt gratitude for her presence and support. Thank you for generously dedicating your time and gracing us with your presence at this event.
Thank you very much for your kind introduction.
Before we begin, I do request all the participants to make a note of instructions carefully. The presenters have to rename themselves as per the allotted codes. The presenters are required to stay online, switch on their camera when the code is called out. Pre-recorded videos will be presented on the screen. Participants cannot reveal their name or any affiliations. Each participant is requested to stay alert for the roll call. All the participants should be present for the session. All the members of the group are allowed to answer the Q&A session. During the Q&A session is limited for 5 minutes.
With this note and permission of the respected judges, let's begin with the oral presentation competition for the undergraduate category, as all our participants and audience are eagerly waiting to present and learn about the new, unique, and interesting ideas that will be presented here. Also, I kindly request the judges to confirm that they have received the scorecard.
Yes, we have received.
Okay, ma'am.
Yes, scorecard is received.
Yes, sir. I would now call upon code 003 to switch on your camera. We will be presenting your video now. We would now call code number 003 to switch on your camera. We will be presenting your video now.
Yes, ma'am, it is on.
I think the student was speaking, but nothing was audible.
Yes, sir, we are looking into the matter.
Yeah. I think our mic is off.
Best carrier for targeted and controlled drug delivery. Greetings to everyone present here. I will be presenting my topic titled as TGM, a smart polymer-based carrier for targeted and controlled drug delivery, as this work focuses on formation of a polymeric network for control and sustained drug delivery system. As being natural mucilage has a greater benefits, being biodegradable, eco-friendly and easily available, but direct use has a certain drawback. To overcome this, we use a green chemistry with the help of method grafted polymerization, which utilizes a synthetic monomer with the help of natural mucilage to develop a smart polymeric network for control and sustained drug delivery. In this, we mainly focused on the Peruvian seeds, which is obtained from, also called as C. alpinia spinosa, obtained from a 50 to 150 herbaceous shrubby plant, mainly found in the areas such as Asia, Africa, Central and South America, and worldwide. Review literature. This overall summary explains us how different seeds have mucilage with the different structure and properties. They also help us to make a pH responsive polymer using a gum arabica. The polymer helps to deliver drug gabapentin effectively in the upper intestine. Also, the graft copolymerization using a starch and acrylamide explains how the polymer form and its characteristic. This overall study inspired us to prepare a new polymer using a Peruvian seeds mucilage. Our aim and objectives, as being natural mucilage, there are some problems as such as hydration issues, microbial contamination, and batch to batch variation. So our project aimed to prepare and test a stable polymer using a period seed mucilage. Objective to synthesize and graft copolymer of a selected polysaccharide using microwave assisted system to characterization of grafted copolymer to conduct an acute toxicity study using OECD guidelines 423 and to evaluate and explore application of a novel polymer structure for control and sustained drug release. Material and method using a microwave assisted system with the help of polyacrylate amide, the grafting of a Peruvian seed mucilage is carried out. For this, 1 gram of Peruvian seed mucilage is taken into the 100 ml of water with the continuous agitation and soaked overnight. By adding a polyacrylate amide and a potassium persulfate to initiate a grafting reaction, the resultant mixture is introduced therefore into the microwave at a 720 watt for 45 seconds. The grafted reaction is left undisturbed overnight to carry grafting process. The acetone is added further for precipitation by repeated washing off with the help of 80 to 20 ratio mixture with the water and methanol. The purified grafted Peruvian seed mucilage is precipitate with the help of formamide and acetic acid with 1:1 ratio and dried at 50 degrees Celsius overnight. We perform all these several batches with the change in the quantity of mucilage and initiator to evaluate the optimal batch. By evaluating its structural and physical properties of the grafted polymers, we come to know that by performing FTIR, the sharp pick of a grafted tarragon seeds or a Peruvian seeds shows that the grafting process has successfully developed. By XRD, we come to know about the amorphous characteristic and crystallinity of the grafted polymers. By scanning electron microscopy, the grafted tarragon and a Peruvian seeds has a porous nature and has a good drug loading capacity. Acute oral toxicity study shows by administering a polymer with the help of minoxidil group of dose with the 200 mg per kg body weight is introduced to the mice and observed 14 days for acute oral toxicity studies. By observation, we come to know that there is no toxic effect after 14 days. Being a good gelling agent of a Peruvian seed mucilage, it is also very beneficial for hair serum that results into the good hair growth in the mice. Summary and conclusion. As to summarize this, we successfully formed a stable polymeric network using a Peruvian seed mucilage. This helps in release of drug for a long duration. Microwave synthetic synthesis technique is being eco-friendly and cost-effective. Toxicity results confirm that the polymer is safe and it may be a good carrier for several drug delivery in further pharmaceutical research. Thank you.
Thank you, code 003. I now invite the judges to ask their questions to the respective presenters.
Okay, so shall I ask or some other judges there? Then she may.
You can carry on.
Yes, sir. Yes, sir, you can.
Yeah. So my first question is that related to that, how many number of trials that you have done?
Yes, sir. Overall, we have performed 7 batches.
Okay.
From that, F4 batch shows the optimum.
Okay. How you came to know that this has been a kind of which kind of the cross-linking, whether there is a linear or a complex, which kind of the cross-linking, how you will come to know?
Sir, we have performed that batches, but we come to know by formation of microbeads, we shows a good effect after administering dose.
In one of the slide where pictures are shown in the drug delivery, you've shown that 2 tablets and one was highly swelled. So are you saying that it will be a highly swellable polymer kind of hydrogel?
Yes, sir. Actually, we have performed that cross-linking with ferric chloride instead of sodium alginate. So it has a good
No, but
binding.
what I understand is that picture that shows that two tablets white color. Okay, so I understand that one is before and one is after the swelling. Is it so?
Yeah.
So
No, sir.
Then?
Sir, it already formulated tablets, sir, that it swell after
You
administration.
have just kept it as a representative that this may happen.
Yes, sir.
Okay. You have mentioned for BCS class 2 and class 4. Any specific, you can say, rationale for that? Why it will beneficial?
Yes, sir.
Why and how?
Can you repeat it, sir?
In your application part, the last concluding slide, you have mentioned about that it is applicable or it will be useful for BCS class 2 and BCS class 4 drugs. So basically a poor drug with a poor solubility. So any specific reason that how it can be helpful for such particular drug?
Yes, after using that polymer, it shows a very slow and sustained drug delivery system after administration. So it will be beneficial for that drug to give a good therapeutic effect.
The class 2 drugs are by default they are sustained because they are poorly soluble.
Yes, sir, but
okay, leave it. Any we can say parameters related to the microwave, what is the dose and how much duration and other things, what are the variations that has been done in that, because it is microwave assisted?
yes, sir, we have
so which kind of the microwave you use, the commonly used in a kitchen or specific one?
Sorry, sir?
The microwave you use which was commonly used in a kitchen, that simpler microwave or any specific microwave synthesizer?
No, sir. That we are using day to day in kitchen. That microscope we used. Microwave.
Because the microwave synthesizer is available whereby you can have the multiple parameter variations.
Yes, sir. No, sir. Day to day microwave we have used.
In general, you have cross-linked with the one, you can say, I think polyacrylamide, right? So
yes,
what is the ratio of your nasal mucilage from that Peruvian seed with the polyacrylamide in your final optimized batch? What was the ratio?
0.02% of acrylamide was taken and 30 ml of mucilage.
No, but finally, if you are having a product as a hard form, right? I think in a dry powder form, you received the final version of the polymer?
No, sir. We have to dry it up. We have to make drying process for that. It is the product form is in semi-solid.
Okay, so still it is in a semi-solid form and you have just used the less than 1% of the polyacrylamide in that.
Yes, sir.
Okay. What do you consider as a novelty? Last question.
Sir, that the Peruvian seed is not having use for polymer formation. That is one novelty, and it show very good sustained release activity after binding with ferric chloride for microbead preparation. And
okay, I think the time is also up. Any other judges there? I think. Ms. Afrin,
Time is up. Can I ask a question?
Yes. I was not aware, sir, ma'am. So that's why I thought I am the only judge.
No, it's fine. Only one question. This acute toxicity studies, how did you do? What did you do? What was the form of dosage?
Ma'am, actually, how did you prepare a microfeed by drug loading, by drug using a minoxidil, and we administered to that mice for under our pharmacology class teacher under her supervision. We have administered the that dose. Okay, yes, ma'am. And we have
Thank you, judges, for your valuable question.
Yes, I have only one question. There are many techniques available for grafting. On what basis this microwave technique which you have used for the current?
Because it takes less time for grafting process. It is a most reason we have taken.
Okay. Okay, thank you.
Thank you, judges, for your valuable question. I now request port 016 to kindly switch on your camera. We will be presenting your video now.
Yes, ma'am, I've switched on my video. Good morning to all the faculty members and respected judges. I am here today to present our research that is titled Design of Cyclodextrin-based mucoadhesive gel for dry socket management, and my slot number is UG16, UG016. So what is dry socket? Dry socket or alveolar osteitis is a painful condition that occurs when the tooth is extracted by the dentist and a blood clot is normally formed, which heals this. But there is disorption of this blood clot and it is disrupted. So this causes pain to the patient, and the current treatment for this issue is normally the paste or irrigation, which has poor adhesion and retention problems. The doctors also offer antibiotics and NSAIDs, but again, this is this causes antimicrobial resistance, and NSAIDs cause digestive issues. So we have eugenol, which is a known analgesic, but again, this also has challenges, that is poor aqueous solubility, which limits its formulation efficacy, and poor retention in the oral cavity due to volatility, which leads to rapid drug loss and short-lived release. Short residence time on the application site leads to intermittent pain relief, and there is bitter taste, which causes patient non-compliance. Moving on, the project or objective was all set to overcome all these limitations, and there is no such formulation available in the Indian market, and no one has focused on it. So our strategy is twofold. We created an inclusion complex using hydroxypropyl beta cyclodextrin to enhance the drug's solubility and stability. We embedded this complex into a mucoadhesive matrix to physically anchor the drug to the dry socket, ensuring prolonged action. Our methodology began with the complexation phase. We chose the kneading method for preparing the eugenol cyclodextrin complex. We used phase solubility studies to accurately determine this optimal component ratio. We then formulated the final gel using this complex combined with mucoadhesive polymers like guaiacol and used carbopol gel, carbopol 934P, for enhancing the viscosity. Our evaluation finally covered our evaluation parameters that are covered were FTIR, DSC, and UV spectroscopy for structurally verifying. Our evaluation focused on other physiochemical tests, that is rheology, pH, mucoadhesive strength to guarantee retention, in vitro drug release to confirm sustained delivery, antibacterial activity against E. coli to validate the therapeutic value. Moving on, we utilized the FTIR analysis to prove that the drug was successfully incorporated into the cyclodextrin catheter. The individual spectra for pure eugenol and hydroxypropyl beta cyclodextrin show distinct functional groups. It is only used to prove there is no reaction between these both polymers, and hence we are using it as a polymer to coat the drug. So the final gel confirms that the drug is well incorporated with no signs of the chemical incompatibility. This successful complexation is foundation for the enhanced solubility. Moving on, we have the gel characterization. For this, We have physiochemical results for pH and rheological properties, and these were optimized, confirming excellent physical stability and suitability for the topical application. Mucoadhesion proof, our most critical performance metric is the impressive mucoadhesive strength that we obtain, that is 22 grams. This robust adhesion guarantees the necessary prolonged residence time at the wound site, fulfilling a core objective for a locally delivered system. This graph here for in vitro drug release demonstrates the functional control achieved via polymer matrix. The in vitro dissolution profile confirms successful modulation of the initial drug release rate, and this is done by the 12-well plate-based method. Then the smooth control curve proves the polymer matrix effectively controls the initial drug burst, which is paramount for achieving the sustained action. Significant, the resulting sustained release profile ensures therapeutic stability for continuous action over a prolonged period. Now you can see that the eugenol and the cyclodextrin loaded gel shows the most sustained and controlled release. Moving on, we also used antibacterial efficacy and safety to prove that it is safe, and the microbes which are generated on the dry socket sites can also be controlled. So the zone of inhibition assay confirms the final product, and it is effective against E. coli. This verifies that the formulation maintained the drug therapeutic potency. Safety assurance, it critically passed the final product, successfully passing the antimicrobial and sterility testing. The optimized gel is now potent, effective against infection, and safe for clinical application. In summary and concluding this, we have successfully designed and validated this novel mucoadhesive system. Our achievements are that we have resolved the eugenol issues, solubility and volatility issues both, and then we have achieved superior mucoadhesive strength of 22 grams, demonstrated a sustained and controlled release drug profile, and confirmed the efficacy and safety by antimicrobial test. For future scope, the essential next step is for evaluation to confirm the clinical efficacy, through which we will be using animal models, and more specifically, we will be focusing on wound healing models and cytotoxicities test. The mucoadhesive gel represents a high promising, novel, and cost-effective local delivery system for dry socket management. Thank you so much.
Thank you, code number 016. I now invite the judges to ask their question to the respective presenters.
Yeah, ma'am, you can ask first, ma'am.
May I?
Yeah.
Yes.
What is the proportion of cyclodextrin and eugenol
It's a
ratio?
1:1 ratio we are maintaining for this.
Okay. 1:1. So was it free flowing like in this proportion, like how it was once you mix it up after kneading and all?
We used kneading method, so it formed a good complex polymer, and it was stable at that time, which further enabled us to go for the further steps.
How do you check the stability? How do you confirm it is stable? No,
Well,
what
there is no
properties did you check it for?
Yes, pardon me.
You said 1:1 is the best combination, no? The proportion which you have
Yes,
taken.
yes.
So how did you confirm it through the quality check? What quality check did you do it to confirm it?
We used FTIR and also certain solubility studies to ensure that this polymer was not, was not affecting our drug product also, and was not even, being, disrupted in our gel formulation.
Okay.
Yes.
Over to you, sir. Daivat sir, you can ask.
Yeah.
Your
Okay.
question.
So you mentioned somewhere about sterility test, and then I think it was related to the antimicrobial test. So there are two different tests, or you're saying that what was the action done was as a sterility test?
Yes.
Okay, so just to make your concept clear, study test is different, and that is what you have done is a cultural sensitivity test. That drug is acting upon.
It's an antimicrobial test
Yes,
to
it is an
ensure that,
antibiotic.
yes,
Study
to
test is
ensure
different.
that the
The
try
product is
socket
sterile or
doesn't
not.
have, okay.
Okay. Again, the same question as of ma'am. That ratio that you have taken is a weight by weight ratio or a molar ratio? 1 mole of drug with a 1 mole of cyclodextrin, or it is weight by weight ratio? Both weight.
Weight by weight ratio, because we are using kneading method hence thereafter. So weight by weight ratio.
Okay. In case of the adhesion study, which method that you have used, can you slightly elaborate, because you have just mentioned the method and the value?
Excuse me, once more, please.
Adhesion, mucoadhesion.
Mucoadhesion. Okay, we used triple beam balance
Okay.
to check the mucoadhesive strength. So we used a porcine mucosa, which was readily available to us, and then
Okay. So
using the
it was
triple
a kind
beam
of a
balance.
ex vivo, right?
Yes.
Okay. Are you aware about any other equipment that can be used? Sophisticated.
Yes, there is a texture analyzer or profile projector that can be used to easily,
Okay.
yes.
Yes. What is the, you can say this, I think you have not shown the solubility data of drug alone, and then what improvement after the cyclodextrin.
Yes.
So because
We
when
just
you are seeing in a
showcase the
conclusion that the
data there.
solubility and permeability, both are increasing, then at least in your data that should be shown that what is the original solubility, and then by the cyclodextrin how much it has been increased.
Yes. We stated the data, that's it, for because we wanted to focus on the FTIR studies then.
Okay. Yes, Rashmi ma'am.
Hello. Yeah. So
Yes,
what did you choose guar gum as a mucoadhesive polymer?
Yes, guar gum is the mucoadhesive polymer choose.
Yeah, but there are other polymers also, no? So specifically, any reason why you used guar gum?
Because this is readily available and it shows more efficacy. That's why.
Did you do any other batches with any other excipient? Any other polymer?
No, we just focused on guar gum.
Okay, fine. Yeah, that's it from my side. Students.
Thank you.
Thank you, judges, for your valuable questions. I would like to call upon code number 019 to switch on your camera. We will be presenting your video now.
Greetings, everyone. I'm excited to share. Actually, there is some issue. I'm unable to turn on my camera. Greetings, everyone. I'm excited to share our work on computational approach to tackle non-alcoholic steatohepatitis or NASH. The title of our presentation is AI-guided network polypharmacotherapy for NASH. In silico identification of novel multipotent herbal leads from Glycyrrhizia glabria. Excuse me. Actually, I'm unable to turn on my camera. There is some issue. Can I rejoin?
Yes, you can do that.
Yeah, thank you so much.
Yeah, we are playing your video. Meanwhile, please quickly rejoin.
Yeah.
Yes.
Greetings, everyone. I'm excited to share our work on computational approach to tackle non-alcoholic steatohepatitis or NASH. The title of our presentation is AI-guided network polypharmacotherapy for NASH. In silico identification of novel multipotent herbal leads from Glycyrrhizia glabria or liqueuris. The core of our study is leveraging AI and network pharmacology to discover multi-target drugs, a strategy known as polypharmacotherapy. This is to address the complexities of NASH. Our focus landed on a compound called glabridin, derived from liqueuris. To understand the need of our approach, let's look at the pathology of NASH. NASH is a severe form of non-alcoholic fatty liver disease. Crucially, it is not a single issue disease. It is defined by three main interconnected pathological features: steatosis, fatty liver; hepatitis, that is inflammation; fibrosis, which can lead to cirrhosis, liver failure, and even cancer. Given the interwoven complexity, we used topological analysis. Through topological analysis, we prioritized two important regulatory points of the disease. These are two critical hub proteins: TNF alpha, the central to inflammation and apoptosis, and PPAR gamma, a master regulator of lipid metabolism and insulin sensitivity. NASH, the failure of monotherapy. This brings us to the central problem of NASH treatment. NASH has a complex, multifactorial etiology. The limitation of conventional therapy is that single target drugs have proven largely ineffective. They fail because they address only one piece of a complex puzzle, leaving the other intertwined pathways to continue driving the disease and making it more severe. Therefore, our rationale for polypharmacotherapy as a multi-target approach is absolutely essential to effectively and systematically manage the disease. We must hit multiple targets simultaneously. The current standard of care primarily relies on lifestyle changes, supportive medication, while the FDA-approved drug we have is resmetirom for non-cirrhotic patients with moderate to advanced fibrosis. We need to innovate multipotent therapies. The research question and study objectives are clearly mentioned. Our study offers a rational multi-target strategy to overcome the inadequacy of single-target drugs. This leads to our primary research question: can AI identify a dual inhibitory NASH drug from Glacyrrhizia glabria liqueurs, targeting both TNF alpha and PPAR gamma? We have three clear objectives to answer this. First one being network mapping. To construct a comprehensive NASH network to identify those principal regulatory nodes like TNF alpha, PPAR gamma. AI screening. To apply machine learning based QSAR and ADMET or ADMET algorithms to filter liquorous phytochemicals for both efficacy and safety. We will see that in further slide. Then molecular docking. Stimulate the dual target engagement to quantify binding affinity against the prioritized hub genes, which are TNF alpha and PPAR gamma. Coming to phytochemical filtration. So our methodology began from phytochemical filtration, a rigorous phytochemical filtration, because our initial library contained over 150 phytochemicals. We passed them through filters, first one being QSAR, to predict the activity against the metabolic targets. Second one was ADMET, screening, a machine learning step for toxicity and drug likeliness. This rigorous funneling successfully reduced the candidate pool from 150 plus to 35 structurally promising non-toxic entities. These 35 entities were then moved to the next step, which is molecular docking. We used industry standard software, Autodock Winner, to stimulate binding against the crystal structures of our two hub targets, TNF alpha and PPAR gamma. Our key metric was binding affinity in kilocal per mole, where a more negative value indicates a stronger favorable binding. Comparative analysis. The ultimate goal of our comparative analysis was to identify candidates that could outperform the standard monotherapies in a stimulated dual target engagement contest. In a sense, we were looking for a single herbal compound that could simultaneously address both inflammation and metabolism dearrangement. Key findings. Our network mapping successfully isolated 20 critical regulatory nodes of NASH. This provided the precise therapeutic map for our drug discovery effort. I wanted to draw your attention to the most important result, summarized in the bar chart. The chart compares the binding affinity of our top herbal lead, glabridin, in the dark blue bar, against a standard reference agent, the lighter blue bar. For both the target hubs, PPR gamma for metabolism and TNF alpha for inflammation. The results strongly suggest that glabridin robustly positioned as stronger dual inhibitory candidate for NASH, capable of simultaneously targeting both the metabolism and the inflammatory targets or the pathologies of the disease. In conclusion, our study validates that an AI-guided network pharmacotherapy pipeline is highly proficient in accelerating the discovery of multi-targeted herbal drugs. The impact is that this approach offers a rational and effective pathway to overcome pervasive limitations of single-target NASH therapies. And most importantly, our key finding is that glabridin is a prime candidate for dual inhibition of TNF alpha and PPAR gamma. Our next crucial step, our future work, is the in vitro and in vivo validation of glabridin's dual inhibitory activity and its overall therapeutic efficacy relevant in NASH models. These are the key references.
Yeah, shall we start? Question answer?
Yes, sure.
Yeah, so it's a good piece of work, especially with the, you can say, in silico method. My question would be like, have you alone done this alone in a manner that the students of the UG level, or there was a support from the other master's, PhD, or faculty?
There was support from faculty.
Okay. So do you at your level understand that what entire procedure was done, the use of software or how the molecular docking and other things that has been done?
Yes, sir. Yes, I do.
So
Because
if any new target that has been given to you and the new molecules set of library are given to you, would you able to check out the whether the binding or the what is the score obtained and how to rely upon that score and other thing?
yes, sir, I can. Obviously with guidance because I'm still learning.
I need guidance. I'm asking that after the completion of this small project, can you do alone without the guidance? That is what my question.
Yes, sir. Yes, sir. Yes, sir.
Okay. Second question is, you have compared with which drug for the last bar chart?
So for TNF alpha, we use thalidomide.
Okay.
And for PPAR gamma, we use rosiglitazone.
Okay. And which is the currently, you can say, the latest drug available or recently approved from the India for this?
So it is not approved from India. It is FDA approved, but it is sold in India as well. It is resmetirom.
Okay. Any drug you are aware about with the from the Zydus?
No, sir.
So just search about the saroglitazar, which is the latest drug for the NASH that they have got several approvals and other things. So you can, in your future study, you can just compare with that. Okay, it is mainly for this disease and liver failure, the non-alcoholic liver failure.
Yes,
okay.
Yes.
Yeah, over to other ma'am.
How do you validate this data?
So ma'am, for validation, we used for network validation and analysis, we use Cytoscape. There are certain guidelines given by that. And again, we for the phytochemical consideration and validation of that, we used the screening by Lipinski's rule of 5, then GIT absorbance, non-hepatotoxicity, HERG inhibition, severity 450 inhibition. We considered these and in the criteria given by CytoHub and then CytoHub plugin by Cytoscape.
Okay. Okay, you can. No questions from my side.
Okay, so one more question from my side is at the end, what is the ratio that you are finding? Because you have taken the herbal drug and from the licorice or specific phytoconstituent that you are planning. So what will be the ratio that will be a kind of a dose that what is expected currently because you have compared it to with the other drug?
Yeah, so we have not yet finalized
No, expected.
Sorry, sir, I cannot answer because we have not reached that level yet.
Okay.
We have just
And
did it
in
in
starting you have
silico.
mentioned with the multi. Okay, so any other drug also like you have already shortlisted from 30 from 150 to 35 to 20. So why only one that you finalize one? Whether the score was relevant with the other two or three more, and we can plan for a polyherbal formulation.
Yes, sir, there was. Actually, we are in process of screening of other drugs as well from Glycyrrhizia glabria only. These were Liquitin, then Vagunin,
No,
Camelic.
I'm asking about the other herbal formulation, other herbal source rather than only licorice.
No, sir, we focused on licorice, so we did not check further.
Okay, thank you.
Yes.
But why specifically licorice, if I can ask?
Actually, we checked the literature and we got a link that licorice offers some drugs which are very potent for NASH network. So we came and we narrowed down our study to licorice.
Okay, fine.
Thank you, ma'am. Thank you, judges.
Thank you, judges, for your valuable questions. I would like to call upon code number 033 to switch on your camera. We'll be presenting your video now.
Good morning, everyone. My undergraduate project code is UG_033. Today, I will be presenting my research on predictive modeling of drug-induced liver injury via machine learning and ADMET profiling. My primary goal in this project was to use advanced computational methods to solve one of the most critical safety issues in modern medicine. Introduction. Let's begin by understanding the gravity of the problem we are facing. First, we have the clinical burden. Drug-induced liver injury, or DILI, is not just a minor side effect. It remains the primary cause of acute liver failure in both the United States and Europe, which possess severe risks to patient safety. Second, this is a massive issue for the pharmaceutical industry regarding attrition. DILI is currently a leading reason for the withdrawal of approved drugs from the market. It is also a primary cause for the termination of promising compounds during late-stage clinical trials, costing billions of dollars. Additionally, limitations of current models. You might wonder, why is this still happening if we have testing procedures? The answer lies in the significant limitations of our current models. For one, in vivo animals models often fail to translate to human toxicity. This is due to species-specific metabolic differences. Basically, a rat's liver does not always process chemicals the same way a human liver does. Furthermore, conventional laboratory assays are resource-heavy, very expensive, and have low throughput, meaning we can't test thousands of drugs quickly. This creates a clear unmet need. We must develop throughput in silico screening tools to predict hepatotoxic liability before a drug is ever synthesized. My study proposes a paradigm shift towards in silico toxicology. The objective was to engineer a robust, gold standard computational classifier that can identify DLL risk prospectively. This approach stands on the three major pillars. First is ethical compliance. It aligns with the three R's: principle, replacement, reduction, and refinement to minimize our reliance on animal testing. Second is the economic strategy. It enables a fair, early, fail cheap approach. By filtering out high risk molecules during the lead optimization phase, we prevent costly late stage failures. Third is mechanistic understanding. We move beyond simple black box predictions to actually understand the physiochemical drivers that cause liver injury. Starting with methodology, data curation. Moving on to how we actually build this model, we started by constructing a gold standard dataset. We curated 1,800 structurally diverse small molecules and performed a binary classification based on their confirmed clinical DILI status, labeling them as either toxic or non toxic. To analyze these molecules, we calculated a multidimensional descriptor space consisting of 150 unique features. These features include physiochemical properties like LOGP and molecular weight, topological indices to measure structural complexity, and quantum chemical properties like HOMO, LUMO gaps to look at reactivity. Crucially, we also included ADMET linked parameters to simulate how the drug is absorbed, distributed, metabolized, and excreted by the body. Methodology algorithmic framework. For a machine learning architecture, we selected a random forest classifier. We choose random forest specifically because it is excellent at handling high dimensional data and nonlinear relationships, and it resists overfitting much better than single decision trees. To ensure our results were reliable, we used stratified tenfold cross validation, which ensures class balance is maintained across all fields. Most importantly, we used a validation strategy that involved a completely independent test set. This allows us to assess the model's generalization capability on unseen data. Our primary focus here was on minimizing type 2 errors or false negatives because missing a toxic drug is the most dangerous error. We showed exceptional discriminative power. We achieved an area under the curve or AUC of 0.94. This indicates that the model is extremely good at distinguishing between hepatotoxic and non-toxic compounds. In terms of predictive accuracy, we reached 89.2% on the independent test set. However, the most critical metric for safety is sensitivity. We achieved a sensitivity of 85.5%. This is paramount in toxicology because it ensures that toxic drugs are flagged effectively, avoiding the worst case scenario of approving a harmful drug. Results and mechanistic interpretation. We didn't just want good numbers, we wanted to understand the biology. By deconvoluting the random forest model and analyzing feature importance, we revealed the key drivers of toxicity. We identified two primary predictors. First is lipophilicity. We found a strong positive correlation with DLL risk. Essentially, highly lipophilic drugs tend to accumulate in hepatic tissue. Second is microsomal clearance. This showed that metabolic stability in human liver microsomes is a critical feature. This confirms the physiological implication that toxicity is driven by the interplay of drug accumulation, lipophilicity, and the metabolic burdens placed on the liver. Discussion. The translational impact of the study on drug discovery is significant. Firstly, this model serves as a deployable ADMET filter that can be used immediately for in-house screening. Second, it provides utility for lead optimization. It gives medicinal chemists clear guidance to modify lipophilicity or metabolic stability to design out toxicity before synthesis. Third, it demonstrates a synergistic approach, proving that combining machine learning with deep ADME profiling yields superior predictive power compared to just using structural alerts alone. Finally, the cost-benefit analysis shows a substantial reduction in the preclinical timeline and budget by prioritizing safer candidates early on. Moving to the conclusion. In conclusion, let me summarize our findings. We have successfully validated a high-performance predictive model with an EUC of 0.94 for DLI risk assessment. We established a clear mechanistic link between specific ADMET properties, specifically lipophilicity and clearance, and hepatotoxicity. This tool provides a rapid, ethical, and cost-effective alternative to early-stage animal testing. A final verdict is that this computational filter significantly enhances the safety profile and efficiency of the drug discovery pipeline. Moving on to references, these are the references used in the study, covering the background of DLA impact, the gold standard data context, and the machine learning methodologies we employ. Thank you for your time and attention. I am now open to any questions you may have.
Thank you, code number 033. I now invite the judges to ask their question to the respective presenter.
Okay, so my first question would be related to the why random forest and not other. Do you have actual idea? Have you done with the
actually, random forest is chosen because it is a robust and versatile machine learning algorithm, and it reduces. Sorry,
which are the other available options?
Other available options are support vector machines, then gradient boosting machines, neural networks, and then logistic regression is simple and interpretable.
For your training data set, how many drugs you have tried?
Sorry, sir?
Training data, because I think it's somewhere you have shown 1800.
Yeah.
So that was a total number of, you can say, the molecules that you explored into the random forest, or it was
yes,
divided
sir, that
in
is the total number of, that is the total number.
Okay, so if you bifurcate training and testing, that how much?
So it is around 1800 to 2000 drug molecules, but the standard is 1800 drug molecules.
No, my question is, you have developed a model, so few you might have used for the training and rest, because you have said that it is validated with a very high, you can say, the reliability results. So for testing, you might have kept some reserve. So is what whether it was done in that way or all you have used for the training purpose?
So all I have used for the training purpose.
Because then I think you should understand that how the ML to be operated, that you cannot do all. Otherwise, it is going to give the 100% or very high result, because it is a memorizing rather than a prediction.
Oh.
You have to keep some reserve. Okay, there is a ratio generally 60-70% that you use initially. Then you do the internal prediction with the which is already used, and then you do the external which is not used, and then you calculate the further regression and other thing, that prediction power. Because if you give everything to the computer and then from that only you are testing that whether what they are predicting, so obviously it will not be a prediction, it will be a memory. So they will give us the same result what you have fed it.
Okay, correct.
Any other drug or molecule other than that have you tested something, because you are saying that it will be helpful, something like that? So any additional done, or it is 1800 which is there?
No, sir, there's no addition done as such until now.
Okay, on what basis that you have out of this selected 1800, how many percentage will were having the hepatotoxicity versus non-hepatotoxic approx ratio?
Approx ratio, so it is for around 30 to 70%.
So 30 were having a hepatotoxicity, 70 were not having a hepatotoxicity.
Yes,
molecules.
Yes.
Okay. Yeah. And whether it was only one, you can say, dependent parameter, hepatotoxicity, yes or no, or any other?
It was, so no, it was just one hepatotoxic parameter.
It was qualitative, yes or no?
That was a primary dependent parameter used in the model.
So, and it was a qualitative, yes or no, or rather 0-1?
It was.
Okay. And for the independent, how many you choose?
So for the independent, I have chosen
total number.
Total number.
Yes, total number of columns, independent parameters.
So
Like
for
log P
everybody-
and lumo, all that thing that you said. So total,
Yes.
how many were the entry points from the gold standard that you obtained? Columns.
So there were around 9 to 10 points obtained.
Okay.
Yes.
That's all from my side. Yeah.
I have a question. One of your strong concluding point is lipophilic drugs will have a high tendency to accumulate in the hepatic tissues. Was there any concluding remark with respect to the log P value? Particular log P value. You said highly
Yes.
lipophilic drugs.
Yes, ma'am. Actually,
Any specific log P value?
yes, ma'am. As lipophilicity is a key factor in understanding drug accumulation, so higher log P values indicate greater lipophilicity, meaning the drug is more likely to dissolve in fats, and the drugs with high log P values tend to have a higher potential for lipotoxicity.
What is a log P value here? We understand a high log P value means what is a numerical which will conclude with this?
Ma'am, actually, the value is greater than 3 is considered to indicate high lipophilicity, meaning the drug can accumulate in the hepatic tissue.
Anything which is more than 3.
Yeah, anything which is greater than 3 is considered to indicate high lipophilicity.
Okay. That's it from my side.
We can conclude.
Thank you, judges, for your valuable question. I would call upon code number 036 to switch your camera. We will be presenting your video now.
Yeah, just a minute. 1027 was absent, right? Breath analysis.
Yes, sir.
Okay, so should we mark absent, right?
So we will be taking it the last.
Okay. Then we'll take it later. Yeah.
Yeah.
Nice.
Code 036, please switch your camera on. We will be presenting your video now.
Good morning. So my title is Blending Network Pharmacology with Indian Knowledge System, Calendula officinalis as a natural remedy for nodular acne. The problem is nodular acne. Nodular acne is a severe type of acne. Severe nodular acne can result in permanent scarring if left untreated. It causes hard lumps or nodules to develop deep under the skin. They are painful and they are very common problem in teenagers. A bacteria called Cutibacterium acnes becomes trapped under your skin in these nodules, leading to infection and inflammation. Without treatment, this type of pimple can lead to permanent and severe scarring. Acne nodules are not treatable with over-the-counter medications alone and might remain for weeks or months. This figure shows the formation of nodular acne. So to treat nodular acne, I choose Calendula officinalis flowers. Calendula officinalis is also known as spot marigold. It is widely cultivated and can be grown in sunny locations. It is an annual or biennial plant attaining a height of 30 to 60 centimeter. Calendula oil is a powerful medicinal oil extracted from the petals of a common species of marigold. Calendula officinalis has been extensively used in homeopathic drug for the treatment of numerous conditions. It shows antimicrobial, antifungal properties. So, pathophysiology of nodular acne. Acne is a chronic inflammatory disease of the pilosebaceous unit. Recent research has shed some new light on the involvement of sebaceous gland as well as on the pro-inflammatory activity of the cutaneous microbiome. During puberty, alteration of the sebaceous lipid profile called stress, irritation, cosmetics and potential dietary factors lead to inflammation and formation of different types of acne lesions. The process leading to a disturbed skin barrier and disequilibrium of the cutaneous microbiome resulting in the proliferation of acne strains is another important process that triggers acne acne activates the nodular acne innate immunity via the expression of protease, activated interleukins, TNF, matrix metalloproteinesis by keratinocytes, resulting in the hyperkeratinization of the pilosebaceous unit. This figure shows the pathophysiology of acne. Calendula officinalis against nodular acne by network pharmacology. Targets of nodular acne and Calendula officinalis are predicted using Swiss target prediction and drug bank. Both target lists are compared by Venn analysis to find common targets. These overlapping targets undergo protein-protein interaction analysis. The PPI network is visualized in Cytoscape software. CytoHubba plugin is used to identify the top 10 key targets involved. A total 119 targets were identified for Calendula officinalis using Swiss target prediction, 102, and drug bank 17 targets. Acne-related genes were retrieved from gene card databases with a total of 19,660 genes related to various types of nodular acne. Formation of Venn diagram. Focus on the genes that appear in the intersection area of diagram, as these are the most likely to be relevant therapeutic targets for Calendula officinalis against nodular acne. These are the Venn diagram, which shows the interaction of common genes. Then formation of PPI interaction network by string database software. The PPI network helps you understand how the common genes identified from the Venn diagram interact with each other at the protein level. The common genes obtained from the interactions in the Venn diagram were then submitted into the string database using the multiple protein section with the organism set to Homo sapiens. After submission, the results were obtained in the form of PPI network, which illustrates the interactions among the common genes. This is the PPI interaction network. Then in the Excel sheet, the data was filtered by applying a filter of 0.4 on the combined score column, allowing for the selection of interactions with a confidence score of 0.4 or higher. Formation network in Cytoscape. The above Excel file obtained from string database after filtering is imported into the Cytoscape, and a network is formed directly as nodes, internodes, and edges were set already. This is the networks of genes that are retrieved through string database. Sorting of top 10 genes by their degree and with CytoHubba in Cytoscape. After the network is formed, the top 10 genes are obtained by the help of CytoHubba. By setting it to calculate mode and setting Hubba nodes the degree mode, information is submitted by ticking shortest path options. After submission, it gives the top 10 ranked genes from the above clustered network of genes with their degree scores. These are the top 10 ranked genes given by the CytoHubba. The following genes are TNF, IL-6, MMP9, PPARG, IL-1B, TLR4, AKT1, EGFR. The figure which shows inflammatory and antimicrobial properties in acne vulgaris. Multiple interconnected innate and adaptive immune pathways contribute to skin inflammation associated with acne. Cutibacterium acne is the predominant organism in the sebaceous region of the skin. Formulation and evaluation of Calendula officinalis face mist for nodular acne. A face mist with its liquid texture is designed to be sprayed onto the skin, providing a rapid burst of hydration and moisture. Its micro-sized droplets settle on the face, delivering a refreshing sensation. This scented water not only hydrates but also offers antioxidants and a healthy glow to the skin. The ingredients which is used to formulate the face mist is Calendula essential oil, witch hazel, polysorbate glycerine, polyvinyl pyrrolidone, rose water, distilled water. Significance and uses of ingredients: Calendula officinalis essential oil. Calendula oil possesses antifungal and antibacterial properties which can aid in preventing and treating infections including acne and wounds. Witch hazel, commonly used for its skin soothing and tightening properties. Polysorbate 80, also known as Tween 80, is a non-ionic surfactant and emulsifier. Rose water is traditionally known as Gulabi Niran. Rose water acts as natural hydrator, providing moisture and helping skin retain it. Glycerine is one of the most commonly used substances in cosmetics. Additionally, it is a key component of moisturizer and lotion. Then PVP. PVP is polyvinyl pyrrolidone, is a non-ionic polymer. Especially in pharmacy, it is used as a binder, coating agent, suspending agent, stabilizer, etc. Distilled water, when used in a face mist, distilled water offers several benefits: hydration, enhanced absorption, and refreshing. The table shows the quantity of ingredients used in the preparation of formulation. Calendula officinalis oil 15 ml, witch hazel 2.5 ml, polysorbate 3 ml, glycerine 2.5 ml, PVP 2 gram, rose water 1.5 ml, and the distilled water is 50 ml. The method of preparation: first of all, ensure clean equipment and containers. Then combine witch hazel and water in a beaker. Then add glycerine. Then add polysorbate and dissolve properly. Why we add polysorbate? Polysorbate act as emulsifier. Then carefully add calendula essential oil, then rose water, and then PVP dissolve in hot water. Then add to the mixture and add distilled water and make up the volume. So we perform different evaluation tests. So the result of the evaluation test is organoleptic properties test. Then result is color milky white, odor woody and musky scent. Skin irritation test, no irritation. pH test 6. Viscosity test 60 millipascal second. Dry time test 2 minutes 10 seconds. Conclusion: the network pharmacology analysis of Calendula officinalis in nodular acne reveals a complex network of interaction between phytochemicals, targets, and pathways. The results suggest that Calendula officinalis may exert its anti-acne effects multiple mechanisms. In summary, this research revealed the potential molecular mechanism of action of Calendula officinalis on nodular acne through network pharmacology. After performing the network pharmacology, we got the top 10 highest degree targets, which are TNF, IL-6, MMP9, PPARG, IL-1B, TLRF. In these top 10 genes, the highest ranked gene is TNF, which has the 43 score by degree method. The formulation and evaluation of Calendula officinalis face mist offer promising solution for safer acne. The calendula's natural properties, including its anti-acne, anti-inflammatory, antimicrobial, and soothing effects on the skin, this product can address various skin concerns effectively. Thorough, rigorous formulation and evaluation process, manufacturer can ensure both efficacy and safety, meeting the growing demand for natural skincare alternatives. Here are some references. Thank you.
Thank you, Code 036. I now invite the judges to ask their questions to the respective presenters.
Okay. So lastly, you have shown one product photograph. So that is your product, or it is just a other, because it was with well labeled marigold. Your mic is off.
Sir, it's my product.
Okay, so is it a part of some startup or something?
Startup.
So your actually the title doesn't match with the work that you have shown, because your title shows only about your initial work, and later on what was done as a part of formulation, that is not covered with the title. So at least you should frame the title appropriately. That is the first comment rather than a question. And then the second one is why you incorporate the term IKS, Indian Knowledge System, into this. Any specific reason for that?
Is because सर नोड्यूलर एक्ने बहुत कॉमन प्रॉब्लम है और उसके लिए सर मैं हिंदी में बोल सकती हूँ ना?
हाँ, बोलो बोलो।
तो सर ये नोड्यूलर एक्ने बहुत कॉमन प्रॉब्लम है और उसके लिए ये बहुत ज्यादा मतलब टीनेजर्स में बहुत ज्यादा है ये प्रॉब्लम, और उसको ट्रीट करने के लिए हम पील थेरेपी कराते हैं, लेजर्स कराते हैं, डर्मेटोलॉजिस्ट के पास जाते हैं, लेकिन वो बहुत कॉस्टली होता है, इसलिए मैंने कैलेंडुला ऑफिसिनालिस एक फ्लावर यूज किया है.
My question is related to the IKS, because when you are using that particular term, तो उसका कुछ relevance होना चाहिए. Throughout your presentation, nowhere IKS came. It was only there in your title.
इंडियन नॉलेज सिस्टम मैंने इसलिए लिया है सर क्योंकि मैंने कैलेंडुला ऑफिसिनालिस ऑयल लिया है फ्लावर से, तो वो फ्लावर हमारे इंडियन नॉलेज सिस्टम बहुत पहले से भी यूज में होता है क्योंकि इट शोज एंटी इन्फ्लेमेटरी प्रॉपर्टीज.
Okay.
ये हमारे गार्लैंड्स में भी यूज
Okay.
होता है.
Okay. Okay. Whatever the work that has been done initially with the network pharmacology and other things, that you have used multiple softwares and other things. So again, the question would be that whether you did it alone or with the support, and if with the support, then if I'm giving you new data, can you do it alone or not?
सर मैंने अपने गाइड के सपोर्ट में किया है, लेकिन मैं सीख चुकी हूँ. मैं अगर कोई नया होगा तो मैं खुद से कर सकती हूँ.
Because these are the complicated softwares, what you are seeing, and what mainly the interpretation, because the data are generated with the you can say the thousands of you can say row. So how to sort, how to interpret, another thing. Whatever the graphs that are generated, they are not easy to understand. That's why my question is. Sure.
सर आई विल डू इट. मैं कर सकती हूँ सर.
Okay. So finally, what is the when you have mentioned the marigold, which is the main component that was important for the acne treatment? Phytoconstituent.
सर रिपीट कर सकते हैं आप क्वेश्चन?
What was the specific phytoconstituent present in this oil of this marigold, which is
सर फ्लेवोनॉइड्स, कैरोटीनॉइड्स, सैपोनिन्स, ये फाइटोकंस्टिट्यूएंट है उसके.
नहीं, as per the docking or as per this molecular pharmacology study that what you have done, any specific molecule that came to a conclusion? Because oil will have the multiple component into that. Whenever you extract anything from the natural sources, there will be multiple ingredient present.
Yes, sir.
So whether your research has incorporated that part, that any specific kind of the phytoconstituent molecule that was giving the main effect of anti-acne?
उसमें TNF alpha, interleukin 6,
They are not
targets.
From this, I'm asking about a phytoconstituent, the drug coming
Chemical
from
constituent. Chemical constituent.
Carotenoids, flavonoids.
Yes, over from my side, over to other judges.
Kavita madam. Okay, one question is, when you prepared that spray, because it is water based, so was it antifungal, वो कुछ नहीं डाला, कुछ preservative नहीं डाला?
Yes ma'am, मैंने ऐड किया है ना preservative.
Which one? What did you add?
Which hazel oil भी ऐड किया है, antioxidant property show करता है. इसमें मैंने
I'm
PVP
asking about a preservative. Preservative. So you don't need a preservative in your formulation. Is it so?
Sir, glycerine, ma'am,
stability.
Ma'am, ये at room temperature.
Okay, fine. Okay.
You have used PVP in your okay. You have used PVP in your formulation. What is the role of PVP here? Function of PVP?
ये binder agent, suspending agent.
Suspending. What is
Binder
the
agent.
specific role over here in your formulation? It has multiple functions, right?
Ma'am, ये मैंने as a binding agent use किया है PVP in my formulation.
Okay. Then, see, it says spray actually. How about its adhesive properties and all? How did you confirm it? Its retainability over the surface?
Can you repeat your question?
See, it's a spray, no? Like how long it can be retained on the surface, like all those things. Did you check and confirm it? Generally, we would have seen so many gels, all those things available in the market, so that it will sustain at the site of action for a long time. So how about your formulation?
मैम मैंने इसमें इवेल्युएशन टेस्ट किए थे, जिसमें विस्कोसिटी टेस्ट किया था, उसका 60 मिलीपास्कल सेकेंड. तो
Okay, fine.
Thank you, judges, for a valuable question. I would call upon code 039 to switch your camera. We will be presenting your video now.
Hello, can I say something?
Yes, sir.
Me and my co-author, we both have joined this meet. So today I'll be presenting our work titled Engineered Biocompatible Luliconazole Hydrogel for Enhanced Retention, Controlled Release and Antifungal Efficacy in Vaginal Candidiasis. And the product name is VG Cleanse, which is a strong against Candida and gentle on you. So the plan of work, as we have mentioned, the first is selection and procurement of drug and excipient. Second is characterization of drug and excipient. Third is formulation of drug-loaded hydrogel. Fourth is characterization of drug-loaded hydrogel in vivo and ex vivo study. And the last is in vivo evaluation of hydrogel. Moving forward to the problem statements, vaginal candidiasis is mainly caused by Candida albicans and leads to itching, burning, abnormal discharge, and discomfort. It occurs due to disturbance of vaginal flora, decrease in lactobacillus, and increase in vaginal pH, which allow Candida to shift from yeast to invasive hyphal form and penetrate the mucosa. Current vaginal creams and gels have poor retention, leakage, short residence time, and require frequent dosing, which result in incomplete cure and recurrence. To overcome these limitations, a mucoadhesive hydrogel-based delivery system was developed. Hydrogels are 3D hydrophilic polymer network, which excellent water absorption, bioadhesion, and patient comfort. The hydrogel was prepared using graft polymerization, where luliconazole is incorporated in CMC DADMAC grafted hydrogel, a cross-link to form a stable mucoadhesive network suitable for vaginal applications. So coming to the third slide, as rationales, luliconazole's sole limitation in conventional vaginal dose forms such as poor retention, leakage, irritation, rapid washout, which reduce therapeutic activity and require frequent dosing. Luliconazole has a strong antifungal efficacy against Candida albicans, but maintaining an effective drug concentration at the infection site is difficult with creams and ointments. A mucoadhesive hydrogel can improve vaginal retention, ensure the drug remains localized for a prolonged period, and it is not rapidly cleared by vaginal fluid or enzymatic activity. Hydrogels provide a comfort and safe delivery system with localized action, minimum systematic absorption, non-greasy medium, and soothing property that enhance patient comfort and compliance. Suitable and controlled drug release from hydrogels maintain consistent therapeutic levels, improving fungal eradication while reducing dosing frequency, treatment failure, and antifungal resistance. Coming to the fourth slide, in methodology, we begin with 7 ml of distilled water and dissolve in 0.5 cmc at 30 to 40 degrees Celsius to form a clear vision solution. Then the DADMAC, a cationic monomer, was added and the mixture was stirred at 65 degrees Celsius at 300 rpm for 15 minutes to ensure the uniform mixing. MBA 247 mg was introduced and the crosslinker and APS 91 mg was added to initiate radical craft polymerization. Stirring was reduced to gentle mixing to allow network formation. After 50 minutes of each reaction time, the solution transformed into a self-supportive hydrogel. This grafted CMC DADMAC hydrogel is designed to be a mucoadhesive, thermosensitive, a suitable form of sustained release of luliconazole. Coming to the test result, the fifth slide, FTIR spectra is mainly done to verify drug excipient compatibility. In the placebo hydrogel, we can see the characterization polymeric peak around 3400 centimeter inverse for 0 for OH stretching, around 2920 centimeter inverse for CH stretching, around 1600 to 1650 centimeter inverse for C double bond O stretching. This peak confirmed the polymer network without any drug. In the drug-loaded hydrogel, the main peak of luliconazole are still present, 3220 centimeter inverse for NH stretching, around 1700 centimeter inverse for C double bond O stretching, around 1550 to 1600 centimeter inverse for C double bond N stretching, around 1450 centimeter inverse for CC stretching. None of this peak disappear and no new peak are formed. So FTR confirmed that the drug is chemically compatible and successfully incorporated without interaction, maintaining its structure inside the hydrogel. XRD report, in the XRD pattern of pure luliconazole, very sharp and intense peak are seen at specific diffractions angle like 20, 10 degree, 14 degree, 18, 22 degree, and 28 degree. The sharp peak indicates the crystalline nature of the drug. But in the drug-loaded hydrogel, the sharp peak disappear or drastically reduce, and the pattern become broad and diffuse. This clearly means that the drug has converted from crystalline to amorphous, molecularly dispersed form where entrapped inside the hydrogel. This amorphous conversion is beneficial because it improves the solubility and support the sustained release of the drug from the hydrogel. DSC. In the DSC of placebo hydrogel, only the thermal transition of the polymer is seen, no major melting peak. In the DCS of pure ludicolage, a sharp melting endothermic peak is present around 90 to 95 degrees Celsius drug melting point. But in the drug-loaded hydrogel, the melting peak of the drug is either completely absent or reduced, showing the drug in uniformly dispersed within the hydrogel matrix instead of remaining in the crystalline form. So the DSC supports polymeric compatibility and thermal stability of the formulation. In vitro drug release. In the in vitro drug release study, the pure drugs show a rapid burst release more than 70 to 80% within the first few hours, but the ludiconazole hydrogel release the drug slowly and consistently over a long period, showing suitable release behavior. Ex vivo permeation studies carried out on rat skin. In the ex vivo permeation study, the hydrogel shows higher cumulative permeation compared to pure drug and marketed formulation, confirming the drug is not only retained longer but also deliver more effectively and target site. So both release study proves that the long-lasting antifungal effect improve retention and reduce dosing frequency. Coming to the sixth slide, the SEM. SEM gives the surface morphology of the hydrogel from the SEM image, which we can see clearly. We see a high three-dimensional network, porous three-dimensional network structure. The pores are not uniform, they vary in size, which is actually beneficial for diffusion. In our formulation, the pore size range from 18.22 micrometer to 39.36 micrometer and 53.76 micrometer to 115.7 micrometer. These large and small interconnected pores allow the hydrogel to absorb vaginal fluid, swell properly, and release the drug in a controlled manner. So FCM confirmed that the hydrogel has a well-developed porous structure which support both swelling behavior and sustained release of drug. Antifungal study. In the antifungal study against Candida albicans, we compared the zone of inhibition of different samples. The placebo hydrogel showed no inhibition, which is expected because it does not contain drug. The pure drug shows antifungal activity but not very sustained. The market formulation also shows a zone but smaller compared to our formulation. Our luliconazole hydrogel shows the largest zone of inhibition, meaning it has a strong antifungal activity and better efficacy than the marketed product. This result proved that our hydrogel released the drug in an effective concentration and maintained it an infection site for a longer time. QBD report optimization. QBD was used to optimize the formulation by studying how the concentration of monomer, cross-link MPA, and initiator APS affect two responses, entrapment efficacy R1 and drug release R2. The 3D structure plot help us understand the relationship between viable. From the graph, we have shown a balanced concentration of monomer and a cross-linker give the highest entrap efficacy. If the cross-linker is too high, the network becomes very tight and the drug release becomes slow. If the cross-linker is too slow, the gel becomes weak and the drug release too quickly. So QBD helps us to identify the best combination of monomer, MBA, and APS for high entrapment efficacy, control, and sustained drug release. Based on QBD result, we selected that the optimization formulation which have the best overall performance. Coming to the next slide. Vaginal irritation test. This study was performed to check the safety of formulation when applied to the vaginal tissue. As you can see the images, the test was done on 4 groups: saline group, hydrogel encap group, strong NOH group, positive control to produce irritation, pure drug group. Across the different time intervals 0 to 3 hours, the saline group did not show any redness or swelling, mainly normal healthy tissue. The strong NOH developed clear sign of irritation such as redness, swelling, inflammation, which confirm the sensitivity of the test model. The pure drug also show mild to moderate irritation, which indicate that a drug alone may cause irritation if applied without a sustainable drug system. The hydrogel treated group show no visible irritation throughout the study, confirming that the formulation is safe, non-irritant, suitable for vaginal application. So the overall irritation study confirmed that the hydrogel prevent irritation, provide better tissue tolerance than the pure drug. Histopathology study was done to observe the condition of vaginal tissue under the microscope and verify where there is any structural damage occurred after treatment. Saline group show normal vaginal tissue with intact epithelial and clear basement membrane. This represents normal structure. Strong NH group show the several tissue disturbed epithelial inflammation and damaged basement membrane. The hydrogel treated group show intact epithelial layer, no inflammation, no structural damage, almost similar to the saline group. And clearly we provide that the hydrogel is non-toxic, biocompatible, do not harm vaginal tissue even after application. Scope of commercialization. There is a high demand in the women healthcare for acting antifungal product or hydrogen, improve patient compliance, biodegradable and safe polymer, cost-effective industrial scale-up, competitive advance over existing vaginal gel. Thank you.
Thank you, 4039. I now invite the judges to ask their question to the respective presenters.
How would you confirm it? It is an hydroporous gel or a hydrogel or a superporous? Which kind of the gel?
Hello.
Yes.
Well, I guess I have some network issue. I cannot see the whole. The
Okay, I'm asking the another question. Am I audible? Hello?
Yes, you are audible, sir. Yes, you are audible, sir.
Yeah, student, because his mic is off, I think. Hello, your mic is off. Can you hear
Sir,
me? Should I ask a question?
no, I would like to answer the first question.
Yes.
Hello. So because we have done the water swelling test, means it swells in water, and then after some time we have taken it and weighed it. So we can say that the hydrogel retains water. So from that means elasticity as well as the porous nature. Porous nature means the absorption of water
Okay.
can be.
Okay. My next question. My next question. Listen. Is it the this new you can say polymer that has been developed for the first time, or some other trial has been done initially, because you prepared it, you synthesizes, right? Monomer and then the chemical reactions. So is it the first time you are doing, or someone else has also already done in a similar manner with the same API, with the same you can say the not API initiator, same initiator and same monomer?
So we have just incorporated the drug inside the polymer gel.
No, but the
as well
polymer
as
that
we
you
have
are
added
prepared, your method
Yes,
shows that you have prepared the polymer as well. It is not a ready-made.
Yes, because we have added some other excipients to make it to improve its strength and all that stuff.
Whether this entire work has been done as a part of the UG research?
Yes.
Because quantum is too high that initially the chemistry part and then later on the Sutix part, including the QBD and other thing.
Yes, in last year, means when I was in the last year, we received a grant on it. So we just means I would not like to tell the name of the institute which gave us
No
grant.
issue.
So, but to
Okay, the question
in
will not waste time in this. Question is on QBD. Can you explain any one parameter's impact? Anyone out of these four diagram that has been shown, 4-5 diagrams, anyone can you explain? Just verbally.
Okay, sure. So we have mainly QBD. In QBD, I just can you? I just don't remember the.
Anyone. That's why
what can we say?
I have given the liberty that anyone that you can.
Okay, anyone. Sure. Here we have taken this as a means monomer, MBA and APS, and means experiment means efficacy we have studied it, on which the MBA as well as we have
Thank
got
you.
that
Over to
curve.
other judges. Okay, thank you.
What was the design of experiment which was used over here? Experimental design?
Which I think. Hello, am I audible?
Yes. Did you get the question? What experimental design you have used in your current study?
Experimental design. Box bank and design. 3-factor box bank and design. In QBD, na?
What are the 3 factors which you have chosen in the study?
Independent factors we have mainly chosen, that is the polymer concentration, cross-linker concentration as a second factor, as well as drug loading or the solvent content as the third factor, which are independent, while we have taken the dependent factors as viscosity, gel strength, drug release, as well as the swelling index of the gel.
So out of all the factors, which was the most prominent one having an influence on the dependent factors?
All the factors are progressing.
Yes, sir.
Okay, fine.
Hello.
Thank you.
Yeah, you can proceed. No question.
Thank you, judges, for your valuable question. I would now call upon code 049 to switch on your camera. We will be presenting your video now.
Hello, good afternoon. Am I visible? Greetings to everyone present.
Yes, ma'am.
Today, I will be representing my team, UG049, and will be presenting our project titled Formulation, Development, and Optimization of Nanomulsion Spray containing beta leaf oil for oral mucositis. I will be covering the following topics, starting off with the background of oral mucositis. Oral mucositis is one of the most painful and disabling complications of chemotherapy and radiotherapy, especially in head and neck cancers. It leads to severe inflammation, ulceration, erythema, difficulty in eating, swallowing, and speaking. It has a direct and significant impact on the quality of life and the cost of care, and also affects survival of the patients because of the risk of infections. Nearly 100% of patients receiving radiotherapy to the oral cavity develop mucositis, and 20 to 40% of chemotherapy patients are affected. Not only does mucositis cause extreme discomfort, often necessitating opioid analgesia, but it is also associated with increased use of health resources and cost of treatment. The rationale for developing a beta leaf oil nanomulsion spray for oral mucositis is to overcome current treatment limitations by combining the therapeutic benefits of a natural bioactive oil with advanced nanoformulation technology. Several treatments exist, like cryotherapy, low-level laser therapy, benzodiazepine and chlorhexidine mouthwashes, topical analgesics such as lidocaine, corticosteroids, etc. However, they have major limitations in that they only provide symptomatic relief with no action on the underlying inflammation. They only have short duration of action requiring frequent reapplication. They pose side effects such as dry mouth, altered taste, burning sensation, drowsiness, and many of these formulations do not provide targeted, long-lasting mucosal coverage. Thus, there is a strong need for a safer, effective, natural, and patient-friendly alternative. Why did we choose beta leaf oil? Beta leaf oil is rich in eugenol, hydroxychavicol, chavibitol, beta-caryophyllene, etc., all proven to be anti-inflammatory, antimicrobial, antioxidant, and analgesic. These properties directly target the pathology of oral mucositis. However, essential oils have challenges like low solubility, poor bioavailability, chemical instability. Nanomulsions overcome these by increasing solubility and stability, enhancing mucosal penetration, and improving bioavailability of the essential oil. A spray formulation further enhances ease of use, even coverage, and patient compliance. Our aim was to develop an optimized beta leaf nanomulsion oral spray for oral mucositis, and our objectives were to perform the pre-formulation studies and to formulate and optimize nanomulsion ratios, to characterize the formulation and evaluate the stability and stress conditions, as well as to perform the antimicrobial and anti-inflammatory activity of our formulation, and finally to determine the in vitro drug release profile of our formulation. Our methodology followed a systematic development pipeline. The first step was to carry out literature review to determine the formulation gaps and review previous studies on oral mucositis and its treatments. Next was to procure suitable raw materials from authentic sources. Thirdly, we carried out pre-formulation studies for the standardization of active ingredient, characterization using UV and FTIR, calibration curve preparation, drug excipient compatibility assessment. Next was the formulation trials, wherein multiple ratios of the oil with the surfactant were tested, and optimization of the nanomulsion was carried out based on clarity, phase separation, and stability. Then came the preparation of the final optimized nanomulsion formulation by magnetic stirring as well as ultrasonication and droplet size reduction. Finally, characterization and evaluation of the final nanomulsion for its physicochemical parameters, stability and thermal and mechanical stress, as well as in vitro studies. Pre-formulation studies helped us understand the chemical and the physical behavior of beta leaf oil. The beta leaf essential oil that was procured was translucent white in color. We determined the lambda max of eugenol, which showed a maximum absorbance at 280 nanometer confirmed by using UV spectrophotometry. For the preparation of the calibration curve, several concentrations of 5 ppm to 100 ppm of standard eugenol were prepared, and a linear calibration curve was obtained, showing good linearity. FTI spectra of both beta leaf oil as well as the standard eugenol were taken, confirming the characteristic peaks of OH stretch, aromatic C-double-bond-C stretch, and CO stretch. This confirmed the identity and the compatibility of beta leaf oil with excipients. All these studies ensured that the material was stable and suitable for formulation. Coming to the actual formulation development, we determined 3% beta leaf oil to be the effective concentration of treating mucositis. We began with 9 different ratios of essential oil to 20, from 1:1 to 1:10. From these, the 3 most stable batches, 1:4, 1:5, and 1:6, were studied in detail. What we observed was that the low surfactant ratios showed phase separation and high turbidity, and the higher ratios formed clearer, stable emulsions. Thus, the optimized formula was selected to be the 1:6 ratio, 3% beta leaf oil, 18% between 20, and water QS. To prepare the nanomulsion, oil and surfactant were mixed together using magnetic stirring. Mixture was subjected to probe sonication for the droplet size reduction, and the final nanomulsion was transferred to a 360 degree long nozzle spray bottle. This process ensured uniform droplet size and long-term stability. The optimized formulation was evaluated on multiple parameters. pH came to be 6.75, which is ideal for the oral mucosa. Viscosity was determined to be 4.7 per centipoise, which ensures easy sprayability. The particle size was an average of 45.91 nanometers, which ensures good penetration and stability, with a polydispersibility index of 0.153 and Z to potential of minus 8.06 millivolts, which is acceptable stability for non-ionic systems. Then, to evaluate the thermodynamic and kinetic stability of our formulation, we subjected the nanomulsion through the heating cooling cycles, freeze-thaw cycles, as well as centrifugation at both 500 and 1000 RPM, in which it showed no phase separation, no flocculation, or creaming. For its antimicrobial activity, we carried out zone of inhibition test of the marketed formulation versus a standard drug versus our preferred nanomulsion. In this test, the marketed formulation, chlorhexidine gluconate, showed a zone of inhibition of 1.8 centimeters. The standard drug of tetracycline showed a zone of inhibition of 2.4 centimeters, while that of a formulation showed 1.8 to 2 centimeters in increasing concentration. This confirms a strong natural antimicrobial activity due to eugenol. Anti-inflammatory activity. Now, the anti-inflammatory activity was determined using the egg albumin denaturation method, wherein the sample is added to a mix of egg white and phosphate buffer, incubated, and then heated to test its anti-inflammatory activity against standard diclofenac sodium. Diclofenac sodium showed an percentage inhibition of the denaturation of albumin at 94.3%, and our nanoemulsion showed a percentage inhibition of denaturation of albumin at 80%. This proves significant anti-inflammatory activity suitable for mucositis. Then, for the drug release, in vitro drug release of our nanomulsion, we use the dialysis membrane method, in which a sample was placed in the dialysis membrane, which is then placed into a phosphate buffer under suitable conditions. Samples were withdrawn at set time intervals, which are then measured for absorbance. The in vitro release showed a sustained release profile with 15% release in the first 30 minutes and a cumulative release of 43% by 3.5 hours. This ensures both immediate and soothing and prolonged action. To conclude, we successfully developed a stable, effective, nanomulsion-based oral spray. It improved the solubility, stability, and bioavailability of betel leaf oil. It showed strong antimicrobial and anti-inflammatory properties and exhibited excellent stability, optimal particle size, proper pH, and good viscosity. Demonstrated sustained drug release and efficient spray coverage. This formulation has strong potential to become a natural, patient-friendly treatment for oral mucositis with fewer side effects than currently available therapies. These are the references that we use for our project. Thank you for your attention. I would be happy to answer any questions.
Thank you, Code 049. I now invite the judges to ask their questions to the respective presenters.
Greetings to everyone present. Today,
okay, the question from my side is, what was the source of betel leaf oil?
we procured our essential oil from a company known as Abbey Naturals.
Okay, so it was a standardized source.
Yes.
Okay, second question is, you have shown the 10 ratios of the oil to twin, and then you have directly shown that the nano emulsion has been formed by one particular method. So what are the variations? Is there any variations done, or any first trial you got the nano emulsion?
No. We tried out 10 different formulation batches from, we varied the ratios of our betel leaf oil as well as a twin pen from 1:1 to 1:10.
After that was the emulsion formation. I'm asking about the conversion of emulsion to nano emulsion.
No. We use probe sonication to reduce the droplet size.
So what are the variations that has been done for in the probe sonication method? The speed, the time, or any other parameter?
We use the probe sonication for up to 15 to 20 minutes. Rest of the parameters.
Single trial that was done converting emulsion to nano emulsion, or some variations that also been done? I'm asking about the variation in process parameter.
Correct. I am not sure of the actual parameters. I do not recall it right now, but we didn't get it in one go, if that's what you're asking.
Okay, because whenever we are doing any pharmaceutics related to the research, then not only the product parameter, but the process parameters exploring are also important. I'm again surprised with you can say that the nano size range below 50.
That is actually the average. It varied between 10 to 100 nanometer.
Okay, and what are the you can say the lastly, I don't recall the final taste that is done. Any I think somewhere you mentioned about, okay, you mentioned about some penetration. So what is your target? Whether the drug should act locally or it should penetrate and further?
I mean, specifically, since we are talking about oral mucositis, it should be localized to the mucosal fields. But often, because of the lesions that are formed, the drug is not often retained. The currently available treatment options that are available, they do not get retained very well to the site of the lesions. And therefore, with the help of a nanoemulsion, because of the smaller droplet size, there could be a possibility of better penetration into the mucosa itself.
Okay. Yeah, that's all from my side.
Thank you.
That's no questions from my side. Kavita madam. Kavita ma'am.
Did you check the stability of the product? Any stability
We
test?
were only able to do the short-term stability testing using the heating cooling cycle, the freeze-thaw cycle, as well as the centrifugation. So in the heating cooling cycle, we subjected our nanoemulsion formulation to different temperatures. So at 4 degrees Celsius for 48 hours, and then immediately at 48 degrees Celsius for 48 hours, and this was repeated thrice. And after subjecting our formulation to this cycle, we saw no type of phase separation or any type of creaming that was observed. Similarly, then we did the freeze-thaw cycle where it was kept in a freezer at minus 29 degrees Celsius for around 24 hours. And then it was thawed at room temperature and then centrifuged at 10,000 RPM. Again, no phase separation was observed. And similarly for the centrifugation as well at 500 and 1,000 RPM. But we've not done any long-term stability testing.
Then what do you recommend for the storage conditions exactly for your product? Because major concern with the nanoemulsions and all is the stability. So currently for your product, what would be the storage conditions which could be drawn from your current studies?
With the current information that me and my team have obtained from the heating cooling cycles and the freeze-thaw cycles that we have subjected a formulation to, I think an ambient room temperature would suffice. But obviously, long-term stability testing will be required to confirm that it is the normal temperature to be stored at.
Okay, thank you.
Right. Thank you.
Thank you, judges, for your valuable question. I would now call upon code number 050 to switch on your camera. We will be presenting your video now.
Greetings everyone. Our team code is UG_050 and our title is Development of a Nano-Emulsion Based Film Forming Minoxidil Spray for Hair Regrowth in Alopecia areata. Our aim is to develop a nano-emulsion based film forming minoxidil spray for hair regrowth in alopecia areata. Our objective is to design and develop a nano-emulsion based film forming spray of minoxidil suitable for topical application on the scalp. Physical, chemical, and in vitro evaluation of developed nano-emulsion formulation. The background and rationale for novel formulation strategy is as follows. Alopecia areata is an autoimmune condition that causes non-scarring, patchy hair loss. Minoxidil is a standard topical therapy that helps stabilize shedding. Its active form, minoxidil sulfate, opens ATP-sensitive potassium channels in dermal papilla cells, promoting membrane hyperpolarization, cell proliferation, and a longer growth phase. The limitations of conventional minoxidil is as follows. Traditional solutions have poor skin penetration and often leave a visible white cast, lowering efficacy and user compliance. The need for improved delivery is as follows. A better system is required to enhance local penetration, eliminate white cast issue, and offer smoother, more uniform application. The benefits of enhanced formulation is improved penetration and superior user experience. Our preliminary studies included solubility, melting point, and pseudo-ternary phase diagram. Solubility of minoxidil was evaluated in water, methanol, individual surfactants, and in 1:1 mixture of surfactant co-surfactant systems. Solubility in various oils was also assessed to identify suitable components for nanoemulsion formulation. The melting point was found to be 248 degrees Celsius, which confirmed drug purity. The pseudo-ternary phase diagram was constructed using water titration between 80 PEG 400 at 2:1 and 1:1 ratios. Oil and surfactant mixtures were tested at various ratios to identify nanoemulsion regions and optimize the final formulation. These are the UV absorbance and FTIR studies. In UV absorbance, the concentration was found to be 3.64 mg, and FTIR analysis was performed to confirm drug identity and check for possible interactions with excipients. This is the pseudo-ternary diagram. The methodology is as follows. First, we prepared a nanoemulsion preparation. In this, oil phase and surfactant phase was created. The oil phase contained minoxidil and IPM. The surfactant phase contained tween 80 and PG 400. These were mixed. After this, water was added drop-wise and mixed with magnetic stirrer with heat. Film forming spray preparation. PVA solution and HPMC solution were prepared. In PVA solution, hot water was used, and in HPMC solution, distilled water was used. These two solutions were combined and mixed with a glass rod. Then our nanoemulsion based film forming spray was created. And this is our final formulation. These are the placebo batches. Our optimized formula included minoxidil as API, tween 80 as surfactant stabilizer and emulsifier, PEG 400 as co-surfactant penetration enhancer and humectant, IPM as oil phase penetration enhancer, emollient and stabilizer, and millipore water as the aqueous phase. The results and data analysis are as follows. Physical appearance. The nanoemulsion showed a uniform homogeneous appearance with no precipitation or phase separation. pH was found to be within acceptable topical range. The percent transmittance was found to be 97.265%. Zeta size was found to be 23.34 nanometer and PDI was found to be 0.332. The spreadability showed that the formulation spread evenly on the glass surface. The drying time was found to be 2 hours at 55 degrees Celsius. Peelability showed that the formulation was able to peel off easily in one piece without residue. Washability of the film was good as the formulation was removed within 30 seconds using water. Thus, we conclude by saying that the nanoemulsion exhibited excellent clarity, suitable pH, nanoscale droplet size, good spreadability, and formed a smooth film with easy peelability and washability, supporting its effectiveness for minoxidil delivery. Future prospects include enhancing film performance, conducting irritation and clinical studies, and optimizing application properties for improved therapeutic outcomes. These are our references. Thank you.
Thank you, Code 050. I now invite the judges to ask their questions to the respective presenters.
Question from my side is on which surface you casted the film for the testing?
Glass.
Okay. But when you consider a real life scenario, biomimicking condition, it would not be a smooth surface because the hair growth you are talking about. So there will be a patient with a different level of the hair growth. Some might have some hair growth, some may not have the completely bald or so.
Yes, sir. Actually, we haven't done that yet. We are going such as Teflon.
Okay. For the spray pattern, any analysis that has been done?
Spray pattern was uniform.
No, but any qualitative, semi-qualitative or quantitative some analysis?
Not yet, sir.
Okay. How much the drying time was there after the spraying?
2 hours.
2 hours.
55. At 55 degrees, it was 2 hours.
No, I'm asking about once you spray, because you are saying that the product will be a film forming spray and the patient has to apply the spray, and then eventually it should form a film. So it should be the minimum possible time and the solvent, because in your formula, I think the water content was too high. I don't recall exactly the percentage, but that was high. So how much time that it would require as per practically, if you have checked?
So the film will start forming slowly. It will take 2 hours to completely form over it. This will increase the contact time. Hence, increase penetration and better user experience.
Okay, and generally 2 hours is, from my understanding, it is too much for a film forming spray formulation. It would be any
Yes, but the thing is,
any liquid formulation after 2 hours on a room temperature on a body with a body temperature will be slightly getting dried. Only thing is polymer is there, then it will form the film. Otherwise, it will not form the film. But when you are saying a film forming spray, then it should form. You can say 2 hours is, from my point of view, it is slightly higher.
yeah, so in normal creams and everything, we get white cast and oily layer. So film will take time, but it will uniformly form over it and it will cause lesser irritation.
Okay. Because generally in that condition, it will drain off liquid, because it is not the even surface on which you are actually biomimicking condition will not be an even surface. Okay, so if it has not been formed, if it is viscosity is less, then it will drain off. So just keep these points in your mind for your future development. Yeah, that's all from my side.
Minoxidil belongs to which class it is? BCS class drug?
Class 2.
Sure.
Yeah.
Okay, then what type of an emulsion it was? Nano O by W or W by O?
W by O, nano W by O.
Which one?
Nano W by O.
W by O. So what was a novelty for choosing this W by O? Already you are telling it's a BCS class 2 drug. Why not
The novelty
O by
was
W here?
Yeah, I'm sorry, we got confused a bit. It is O by W because water is 67%. And hence we have enhanced solubility using surfactants to one twin 80 and one TEG 400, which also works as co-solvent at the same time.
Okay, how did you disperse the drug? In which phase did you disperse it first?
First, we took the isopropyl myristate, the oil phase.
Okay.
And we used two surfactants in film forming spray. And we used the ultrasonication method. We did not get the RPM at the first try. We tried to optimize it. And the one with the most clarity was taken so that emulsion was not turbid and showed proper stability in terms of how the droplets did not differentiate.
Did you plot the phase diagram here? What software or how it is done?
So the software, we put the data in it.
Sorry?
The data it was a software. We took the data about the surfactant, oil, and water, and we put it in a software. And then
What software it is? Name of the software, right?
I'm not able to recall just a second, ma'am.
Okay, fine. Thank you.
Thank you.
Carry on with the presentation next.
Thank you, judges, for valuable questions. I now invite code number 027 to switch on your camera. We'll be presenting your video now. We are presenting. Please give us a minute.
Hello, I think some network issue was there. May I know who is presenting or?
Code number 027 is present. Just a minute.
Yeah, okay, no problem.
AI app which detect the breathing. Good morning. Today I am going to tell about one app which is so what is respiration? Good morning. Today I am going to tell about one app which is Breathe AI app which detect the breathing rate and analyzes the hidden health clues of respiratory system. So what is respiration? Respiration. The respiratory system is the system in our body that helps us to breathe. It supplies O2 to the body, means oxygen, and removes carbon dioxide from body. Main function of respiration is breathing, gas exchange, oxygen transport, voice production, smell, olfaction, maintaining blood pH. There are some respiratory diseases. First, obstructive lung disease. Airflow is blocked or narrowed. Means in obstructive lung disease, humans airflow is blocked and or narrowed means it become longer, making it hard to exhale. Means in this exhalation is hard for human being. Person can't exhale properly. Example of these diseases are asthma, chronic obstructive pulmonary disease COPD, bronchitis. Now second one is restrictive lung disease. Lungs cannot expand fully, reduce lung volume. In this volume of the lung is reduced. Lungs cannot work properly or do their function properly. It get expand fully. Examples of this is pulmonary fibrosis, sarcoidosis, interstitial lung disease. There are some upper respiratory diseases also, common cold, sinusitis, pharyngitis and laryngitis. Now I will tell you the breathing rate of some respiratory diseases. Then first for normal adult it is 12 to 20. Then second asthma it is 20 to 30. COPD it is 20 to 28. Pneumonia it is 24 to 30. Tuberculosis 20 to 30. COVID-19 22 to 30. Then acute respiratory distress syndrome it is 30 to 40. And in bronchitis in acute it is 18 to 24. In pulmonary embolism it is 28 to 40. In pneumothorax it is 30 to 40. Now I will tell you the problem statement of this is respiratory disease and breathing rates. Respiratory diseases are increasing rapidly, including asthma, COPD. Means some respiratory diseases are increasing rapidly in India, like asthma, COPD. These are chronic obstructive pulmonary diseases, pneumonia, bronchitis, COVID-19, and other infections. Each disease affects breathing rate, which is the most basic and important vital sign. Normal versus disease breathing rates. Normal adult breathing rates is 12 to 20 breath per minute. Asthma attack. In asthma attack, it go higher than 20. Go plus in 20 means it will be 25 to 30 breath per minute. In pneumonia, it is 30 plus breath per minute. In COPD, it is 25 to 35 breath per minute. In bronchitis, it is 20 to 28 breath per minute. Severe respiratory distress, it is more than 40 breath per minute. Tracking breathing rate early can actually predict danger and help prevent hospitalization. Then our solution for this problem statement was one app, AI-powered breathing analysis app. In this hackathon, we built an AI-based breathing monitoring app that helps detect abnormal breathing early. How the app works? First, user opens the app and simply keeps the phone near their chest or face or near nose. Second, the phone's microphone and sensor capture the breathing sound pattern. Third, our AI model analyzes breathing rate, wheezing, crackles, or obstruction sounds irregular patterns. The app then shows normal, warning, or high risk breathing status. The data is saved and can be shared with a doctor. Thank you.
This is the app. Breathe AI, hidden respiratory health clues, real-time automatic breath detection powered by AI, normal adult breathing rate, 12 to 20 breathings per minute. Here we fill patient details, name, gender, and age. Then start measurement mode, 30 seconds. Here we can see breathing rates. By using the sensor, we are able to record the readings, show how it records the breathings. After uploading the readings of breathing per minute through sensor, the app is able to predict the respiratory diseases. Thank you.
Dear judges, we would like to point out that the guidelines for the videos were not followed, so we can disqualify this presentation.
Okay, so there is a need to ask a question or straight away disqualified?
If you want to, you can ask.
But if not to be considered, then why to ask?
Same as with the code 055. So you want to see the video or we can proceed further?
No, it's as per your guideline, whatever we have dedicated 3 hour time for you. So whatever is as per your procedure.
Okay, sir. Thank you.
If it is to be rejected, then no need to waste the time. But if it is a part of the requirement, then we have to. Whatever the ma'am takes a call.
Okay, sir.
Your internal, you can say the ma'am might be from that
Now we request the judges to share a few words about their experience on today's event.
Hello.
Yes, sir, you're audible.
Yeah. So, good afternoon, ma'am. Rashmi and Kavita, I think you both from the same BNCP. I think we are meeting online for the first time.
Sir, I'm from
No, no.
Krupanidhi, sir.
You are from?
Krupanidhi College of Pharmacy, Bangalore.
Okay. So, sorry, I'm not much aware. Sorry, ma'am.
I'm from
Yes, sir.
BNCP actually.
No, I thought that students might have kept some internal and some external. So, and
Yeah. So I am the internal BNCP. I'm from BNCP.
okay. And Kavita ma'am is from Krupanidhi.
Yes, sir. Yes.
Okay. Good to meet you, ma'am.
Same with us, sir. Same with me also.
Yes, so if you want my input as an experience, then yes, it's a good effort that I have been there for the in the first such student research congress when it was started in COVID time. So I was judge at that time as well, and I can see the lot of improvement since this 6-year journey. This is the sixth one. Again, I have been invited for the judge, so a lot of improvement is there, including this. Yeah, and when I see the schedule also, in schedule also it is as the scope has been improved. Earlier it was too narrow, and only the PhD students were there, and a few of the PG students have presented the work. And now the UG students are also actively performing and presenting the work. So yes, the level that has been improved from the you can say the organizer side, it is very good improvement, and especially when you are saying it is for the student and by the students in most of the case, the way the students are handling it. So I must appreciate that because it is very rarely observed, especially in the pharma field. Yes, in technology and engineering, there are several student chapters are that they are actively doing the large scale events, but in pharma it is very rare. Must give the congratulations to the faculty of that you can say the institute BNCP as well as the students. It's really good. And yes, presentation wise also the I think you might have shortlisted also. They almost all were of good work. Only thing is the reason, if you observe that why I was asking since the first question frequently, because generally it happens in an online session that the students keep the chat GPT or other tool open, and then they answer it after what that the chat GPT answers. So that's why I was quickly changing the question. And only one or two students I observed that I believe that he might was taking the help from the some other source to answer the way he was taking the pause and other thing. So that was what I understand that in such online session only you can say we should not give time for the student to respond. Then it's
That's what. So
yeah.
5 minutes was kept only for that. That's it.
Yes.
The question can be rapid fire line.
Right. Yes, Kavita, up to you.
Yes, sir. Like this is my second time. Last time also I got a chance to be one of the evaluator for the same UG program. So I can say it is a very structured program. Our organizers, I must congratulate, online program without any single glitches. Just timings also managed very well. Our anchors, and it was wonderful program. It's always an learning process for us being into this part of the event. So thank you once again. I thank Madam for this special thanks to Madam for inviting us and giving an opportunity to be a great program, ma'am. So it's a great learning for us too. And even my co-evaluator, sir, so like, he was with the list of questions, like, sir was very thorough with the topics, everything. Yes, sir. I too had a very good time. And thanks to Madam also.
Yes.
Yeah.
Thank you, Kavita Madam. Yeah, thank you very much. Thank you, Daivat
how
sir.
are you?
Hello, Daivat sir. How are you? Thank you, Daivat, for joining us.
It was my honor, ma'am.
I know Nirma का बहुत hectic schedule रहता है. Thank you.
Yeah, it was just unplanned meeting came at 11 and it was on, but still I came out of the meeting saying that I have to join over here. So Afrin has called me, but I said that I'll be joining just 2 to 3 minutes prior to the time.
Thank you very much, Dr. Daivat and Dr. Kavita. Thank you, Rashmi Madam, for joining the team.
So actually, these Daivat sir and Kavita madam had a lot of questions. Everything was covered. So if you see, I had asked, so organizing team, what you told, I followed. Questions were limited. And mostly Daivat sir and Kavita madam had put number of questions. So it was
good. So that's the whole idea. See, we wanted students to learn from experts. And when they present, they learn also. So when the questions are being raised, then they get more clarity. So that's great.
Nice questions,
the whole idea was to inspire them to push them to think further, to broaden their horizon of understanding.
And before you join, I was just saying the same thing that when I can say just the first time during the COVID time, and compared to that, if you see there is a lot of improvement from the student side, the way you are organizing the structured way and increasing the scope and other things. So hats off to you as a leader. At the same time, your entire team and the students, those who are motivated towards the, you can say generally it happen that whenever you ask the students to arrange any event or so, they will be more towards the cultural and co-cultural rather than this kind of the extracurricular activities, rather than you can say the research congress. So it's good that actively they are doing a lot of work.
Yeah, they learn a lot during this process.
Yes.
Yeah, thank you very much.
Sir, can you please turn on your camera so we could take a picture?
I'm trying to turn on because, but somehow it might have been blocked from your side or so. I don't know. Because from my side it is on.
It's open.
It is not.
The camera of the laptop or something must be close to it.
I don't know. Because it shows when I you can say drop down the menu, it shows the camera is on. Even options are showing with the background and other things, but it is not clicking it open.
Oh, sir, Nirma has blocked.
Yeah, most of because that
Probably
might be
Nirma
possible
has
because
blocked.
Since a few you can say the last 2 weeks before that they have changed the completely wire firewall, the blockage.
Access.
Even currently for Nipicon also recently started with the third party, and it happens that every time whatever their data storage, other things, we have to request to our IT team to keep it open.
I think that could be the reason.
Yeah, maybe.
Yeah, no problem. All right. Yeah. Over to you, team.
I have shared one photo from my side where my camera is on to Munira ma'am.
No problem.
On your WhatsApp.
Yeah, I see it. Yeah, I see it. Yeah, I see you are very much visible. No problem.
Maybe yes, as you are saying that.
Some issue.
Okay, just let me know let me take the full picture so that at least your banner and other things get covered. At least for your record purpose.
Yeah.
Just a minute.
Sure. Yeah. I shared the screenshot with you.
Thank you, ma'am. I will share with you.
Yeah, okay. Thank you. Thanks.
Thank you. We would like to request you to please share the scorecard with us.
Yeah, so hard copy with signature is required or soft copy would be okay, because you have mentioned signature.
Soft copy would be okay. No, so you can put your scanned signature.
Yeah, digital signature is okay, no?
Yeah, scanned signature is okay.
No problem.
Yeah.
Okay, then mine is ready. I'll just share it in 5 minutes.
Okay, thank you.
Thank you.
On behalf of entire organizing team, it is my honor to deliver the vote of thanks on this wonderful occasion. First and foremost, I extend our deepest gratitude to esteemed judges. Your time, thoughtful evaluation, and insightful comments have added an immense value to our event. Your expertise has not only guided the young researcher, but has also elevated the academic quality of their session. We sincerely appreciate your generous contribution to the success of today's program. A heartfelt appreciation goes out to all the participants of undergraduate category. Your energy, curiosity, and dedication have truly brought this session to life. Each presentation reflected hard work and passion. We are immensely proud of every one of you. Your enthusiasm is what makes this Congress meaningful and memorable. Finally, I would like to thank each member from the audience for your presence, encouragement, and engagement. Your support plays an important role in fostering a vibrant research culture and making events like this impactful. Thank you once again to everyone for being a part of this inspiring experience.
We look forward to seeing you in the upcoming sessions as well. As we conclude the day one of the 6th Student Research Congress 2025, we warmly invite all participants and attendees to join us tomorrow for an exciting day 2, which features the keynote address by Dr. Shubhashish Chakraborty, followed by PhD oral presentation, Hackathon Oral Solids, and Hackathon Artificial Intelligence category. We encourage everyone to participate with the same enthusiasm and spirit of learning. We also look forward to seeing you for the award and valedictory session, where we will celebrate the outstanding effort and achievement of our participants. Thank you
so much.
Thank you very much, everyone.
Thank you, ma'am. Good day. Bye-bye.
Thank you. Thank you too. Bye.
Thank you,
Kavita,
Thank
madam.
you.
Give my
Bye.
regards to Ramesh,
Sure.
dancer. Yes.
Yeah, 100%.
Thank you.
Take care,
Thanks.
ma'am.
Bye. You too. Thank you, everyone.